Close your eyes for a moment, and before you drift too far into the quiet of the night, take a breath. Feel the weight of your body against the mattress, the slow, rhythmic rise and fall of your chest. In this stillness, your body is performing a silent, ancient miracle. Millions of tiny architects are moving, repairing and building within you while you rest. They don't need your conscious direction. They simply know their craft. Tonight, we are going to look at the very foundation of that craft. The quiet, elegant building blocks that make up your muscles, your skin, your enzymes, and even the very thoughts that are currently beginning to soften into dreams. We are embarking on a journey through the 20 amino acids, the alphabet of life itself. It is a beautiful thing to realize that everything you are in your physical form is written in a language of only 20 letters. But before we learn the words, we must understand the paper they are written on. We must understand the core, the anchor, the steady center that holds every amino acid together. This is a journey into the heart of biochemistry. But do not feel you need to strain to listen. Let the concepts wash over you like a gentle tide. If you find this peaceful exploration helpful for your rest and your learning, I invite you to join our quiet community here by subscribing and leaving a gentle mark of your presence. It helps us keep this fire burning for you night after night. But for now, just settle in. Let the world outside fade away. Let us begin where all journeys in biochemistry begin, not with a complex formula, but with a simple, steady atom. Imagine, if you will, a vast, dark ocean. In the center of this ocean, there is a single, glowing point of light. This is a carbon atom. In the world of biology, carbon is the great connector, the ultimate social butterfly of the elemental world. It has 4 hands, 4 ways of reaching out to touch the universe around it. In the structure of an amino acid, we call the central carbon the alpha carbon, or C sub alpha. It is the alpha because it is the primary seat of power, the molecular compass from which all other directions are determined. Everything we discuss tonight, and every night after, will be anchored to this one steady point. The alpha carbon does not stand alone. It reaches out in 4 distinct directions, forming a shape called a tetrahedron. If you can imagine a small pyramid with a triangular base, the carbon sits right in the center, and its 4 hands reach toward the 4 corners. This geometry is not accidental. It is a masterpiece of spatial balance. The first hand, pointing upward like a steadying mast, holds a single hydrogen atom. This is the smallest, simplest element in existence. In our molecular compass, think of this hydrogen as north. It is the constant, the lightweight anchor that provides balance without adding bulk. It is always there, a silent partner in the dance of life. Now, let's look to the left, to the west of our compass. Here, the alpha carbon holds on to the amino group. This is where the name amino acid comes from. The amino group is a small energetic cluster consisting of a single nitrogen atom bonded to 2 or 3 hydrogen atoms, represented as -NH2 or -NH3+. Nitrogen is a fascinating element. It is the breath of life, the element that makes up the majority of the air you are breathing right now. In this group, it carries a slightly positive charge, like a soft magnetic pole. It is basic, or alkaline, in nature. Think of the amino group as a blue, cool mist, a place of potential energy, ready to reach out and form a bond with a neighbor. To the right, to the east of our central carbon, we find the carboxyl group. This is the acid part of the name. It is written as -COOH or -COO-. This group is made of carbon and oxygen, and unlike the cool, basic nitrogen on the other side, this group is acidic. It carries a negative charge, a sharp red ember of reactivity. It is the part of the molecule that is willing to give away a proton, to let go of a piece of itself to facilitate a reaction. This creates a beautiful internal tension within the molecule. On one side, you have the positive, accepting amino group. On the other, the negative, giving carboxyl group. This is the Zwitterian state, a German word meaning hybrid ion. Even within this tiny single molecule, there is a perfect balance of opposites, a microscopic yin and yang that allows it to exist in the watery world of your cells. But we are missing the most important part of the compass. We have the north, the hydrogen, the west, the amino, and the east, the carboxyl. Now, look down. Pointing toward the south is the fourth hand of the alpha carbon. This is the R group, often called the side chain. While every one of the 20 amino acids has the same top, left, and right, the R group is where the magic happens. It is the variable. It is the traveler's backpack. If the alpha carbon is the chassis of a car, the R group determines if that car is a rugged truck, a fast racer, or a steady sedan. It is the R group that gives an amino acid its personality, whether it loves water or fears it, whether it is bulky or tiny, whether it carries a charge or remains neutral. Over the next 19 lessons, we will be exploring these 20 different R groups, seeing how each one adds a unique note to the symphony of your biology. As you drift deeper into relaxation, consider the strange and beautiful concept of chirality. Because the alpha carbon is bonded to 4 different things, it becomes what we call a chiral center. This is a fancy way of saying it has handedness. Place your hands in front of you, or imagine them in your mind's eye. Your left hand and your right hand are mirror images of each other, but they are not identical. You cannot slide a left-hand glove onto your right hand and have it fit perfectly. Molecules are the same. In the vast kingdom of life on Earth, from the smallest bacteria in the soil to the complex neurons in your brain, almost every amino acid used to build proteins is left-handed, or the L isomer. There are right-handed versions in the universe, but life has chosen the left. It is a mystery of the cosmos, a signature of our common ancestry. You are built of trillions of tiny left-handed travelers, all oriented in the same direction, clicking together like perfect gears in a cosmic watch. Think of the alpha carbon as a wise old tree in the center of a clearing. Its roots are the R groups, reaching deep into the soil of chemical possibility. Its branches are the amino and carboxyl groups, reaching out to hold hands with other trees to form the great forest of a protein. And the hydrogen is the wind that moves through the leaves, light and ever-present. This structure is incredibly stable, yet infinitely flexible. It is strong enough to hold the weight of your skeleton, yet delicate enough to fold into the shape of an antibody that protects you from illness. In the quiet of your room, you might wonder why this matters. It matters because this tiny geometry is the reason you can digest your dinner, the reason your heart knows how to beat, and the reason you can dream. Each amino acid is a letter, and the alpha carbon is the ink. Without this specific arrangement, this molecular compass, the story of life would have no way to be written. The nitrogen and the oxygen, the carbon and the hydrogen, they all dance around that central alpha point, waiting for the signal to join together. As we finish this first look at our molecular map, imagine that central carbon glowing a little brighter in your mind. It is steady. It is balanced. It is the foundation. You don't need to memorize the angles or the charges. Your body already knows them. It has been using them since before you were born. Every time you heal a small scratch or grow a new strand of hair, you are watching the alpha carbon at work. It is a quiet, tireless servant of your existence. Let the image of this perfect, four-handed molecule settle into your thoughts. It is symmetrical yet diverse, simple yet profound. It is the start button for every biological process you will ever experience. We have established our center. We have set our compass to north, south, east, and west. We know the players: the hydrogen, the amino, the carboxyl, and the mysterious R group waiting in the wings. And with that understanding quietly in place, we're naturally led to ask, what is the simplest thing a traveler could carry in that R group backpack? If the R group is a space for variety, what happens when we fill it with the simplest, lightest thing possible? That leads us to our first specific amino acid, a tiny minimalist that proves that even the smallest presence can change the world. In our next quiet moment, we will look at the tiny, flexible wonder known as glycine. But for now, just breathe. The compass is set, and the journey has begun. Glycine. In our last quiet session together, we ended with the idea of the R group being like a traveler's luggage, a unique backpack that gives every amino acid its name and its purpose. We looked at that central alpha carbon, that molecular compass, and we saw how it stands ready to hold on to whatever life requires. Now I want you to imagine a traveler who chooses to carry almost nothing at all. This traveler is glycine, the simplest, the smallest, and perhaps the most humble of all the 20 building blocks. While others carry heavy rings or long complex chains of carbon, glycine's side chain is just a single hydrogen atom. Because glycine is so tiny, it possesses a superpower that none of its larger cousins can claim. It is the most flexible of all the amino acids. In the grand, crowded dance of a protein molecule, space is often at a premium. Large amino acids with their bulky packs bump into one another, restricted by their own size. But glycine, with its single hydrogen atom, can tuck into the tightest corners, like a small child finding a perfect hiding spot in a room full of adults. It can rotate and bend in ways that would be physically impossible for any other molecule. It is the molecular hinge that allows proteins to turn corners and fold back on themselves with grace. When we look closer at this tiny structure, we find a fascinating exception to the rule we discussed regarding handedness. You might remember that most amino acids are chiral, meaning they have a left-handed and a right-handed version. This happens because the central alpha carbon is usually bonded to 4 different things. But let's look at glycine. It is bonded to the amino group, the carboxyl group, and 2 hydrogen atoms, one that is part of the standard backbone and one that serves as its R group. Because it has 2 identical partners, glycine loses its handedness. It is achiral. It is the only amino acid that is perfectly symmetrical, a mirror image of itself. In the world of biochemistry, glycine is the point of perfect balance, the neutral center where the distinction between left and right simply fades away. Imagine a massive, intricate tapestry being woven. Most of the threads are thick and colorful, creating the bold patterns and the sturdy fabric. But to make a sharp corner or a delicate loop, the weaver needs a thread that is nearly invisible, yet incredibly strong. That thread is glycine. In our bodies, we see this most clearly in collagen. Collagen is the most abundant protein in your body. It is the glue that holds your skin together, the strength in your tendons, and the framework of your bones. The structure of collagen is like a heavy, three-stranded rope, where three long chains of amino acids are wound tightly around one another in a triple helix. For this rope to be strong, the strands must be packed together with incredible density. There is simply no room for bulky side chains in the center of that coil. To solve this, your body uses a repeating pattern. Every third amino acid in a collagen strand is a glycine. Its tiny hydrogen pack faces inward, allowing the three strands to sit flush against one another. Without the unassuming minimalist nature of glycine, your body would quite literally fall apart. We owe the very integrity of our physical form to the fact that this one molecule was willing to carry the smallest load. But the story of glycine doesn't stop with the structure of your skin. It also plays a quiet, vital role in the way your nervous system communicates. In your spinal cord and the lower parts of your brain, glycine acts as an inhibitory neurotransmitter. Think of your nervous system as a vast network of highways. Some chemicals, like the ones we will discuss later, are like green lights, telling your neurons to fire and move and act. But a system with only green lights would be chaotic and dangerous. We need red lights to bring order, to calm the noise, and to ensure that signals only move when they are supposed to. When glycine binds to its receptors, it opens a tiny gate that allows negatively charged chloride ions to flow into the neuron. This makes the neuron less likely to fire, effectively quieting the electrical storm. It is one of the ways your body maintains its stillness. In fact, as you lie here now, drifting toward sleep, glycine is one of the silent guardians helping your muscles remain relaxed and your mind remain calm. It is the chemical embodiment of a soft hush, whispered across your nerves. It reminds us that in biology, as in life, sometimes the most powerful thing you can do is provide a space of quiet and restraint. Even in the world of metabolism, glycine is a versatile worker. It is used to create heme, the part of your red blood cells that carries oxygen from your lungs to your toes. It is used to make creatine, which provides a burst of energy to your muscles. It even helps your liver detoxify chemicals, acting as a conjugator that grabs onto unwanted molecules and helps escort them out of your body. It is a molecule that is always in high demand, yet it never asks for much in the way of resources or space. It is the ultimate team player, the minimalist architect who builds the foundation so that others can create the flourishes. There is a beautiful lesson in the existence of glycine. We often think that complexity and size are the markers of importance. We look for the biggest, the brightest, and the most intricate. But the alpha carbon's first and simplest creation teaches us otherwise. By carrying almost nothing, glycine becomes the most versatile tool in the toolkit. By being small, It makes the strongest structures possible. By being neutral and symmetrical, it brings balance to the handedness of life. It is a reminder that there is a deep, hidden strength in simplicity. As you breathe in and out, let the idea of glycine settle beside you. Think of the millions of tiny hinges in your body, allowing your proteins to fold and function. Think of the tight, strong ropes of collagen keeping you whole. Think of the quiet, red lights in your spine, allowing you to rest in peace. All of this is made possible by a single hydrogen atom resting on a central carbon. We have seen what happens when the traveler carries nothing. We have seen how a lack of baggage allows for a freedom of movement that is truly extraordinary. But life is rarely content with staying minimal. Evolution likes to build, to add, and to see what happens when we introduce just a little bit more substance to the frame. We have mastered the art of the hinge. Now, we must begin to build the wall. Let's set that idea down now, fully formed, and let our attention drift toward the more complex shapes that these sturdy bricks can take. We have explored the lightest traveler, the one who carries only a single atom of hydrogen. But what if we were to take that same traveler and add just one small sturdy piece of wood to their pack? What if we added a single carbon and a few more hydrogens, creating a small, solid block? So what does this structure actually do when we add just a little bit more substance? That's the question we'll gently explore next, as we look at the steady, reliable foundation of the molecular world, alanine. In our last quiet session together, we ended with the idea of the minimalist traveler, Glycine, who carries nothing but a single hydrogen atom in their pack. We saw how that smallness allowed for a unique kind of freedom, the ability to tuck into tight corners and provide the flexibility that life requires. Let's pick up that thread right where we left it. If Glycine is the minimalist who travels light, our next companion represents the first step toward building something solid and permanent. This is Alanine. Imagine for a moment that we take that same traveler, and instead of a single hydrogen atom, we give them a small, sturdy wooden block to carry. In the language of chemistry, this block is a methyl group, written as -CH3. It consists of one carbon atom holding on to 3 hydrogen atoms. It is the simplest hydrocarbon side chain possible. If glycine was a whisper, alanine is a clear, steady note. It isn't flashy. It doesn't carry an electric charge, and it doesn't reach out to grab other molecules with a sulfur handshake. Instead, it offers something just as vital: reliability. Think of alanine as a smooth, unadorned brick. When you are building a great cathedral, you need the stained glass and the ornate carvings to give the space its beauty. But the cathedral would not stand for a single day without the thousands of simple, uniform bricks that make up the hidden interior of the walls. Alanine is that brick. It is one of the most abundant amino acids in the world of proteins because it is the perfect size for building the structural core. It provides the bulk that allows a protein to take up space and hold its shape against the pressures of the cellular environment. To understand why alanine is so important, we have to talk about its relationship with water. You see, the inside of your cells is a very crowded, watery place. Most of the molecules in your body are polar, meaning they love to dance with water. But alanine is hydrophobic. If you've ever watched rain bead up on the surface of a freshly waxed car, you have seen the hydrophobic effect in action. The water and the wax simply do not want to mix. In our biological world, alanine is like that wax. Because its side chain is made only of carbon and hydrogen, it finds no comfort in the presence of water. When a long chain of amino acids begins to fold into a protein, the alanines all behave like a group of shy people at a crowded party. They don't want to be out on the surface where the water is. Instead, they huddle together in the very center of the protein. They move inward, pushing the water away, and in doing so, they create a dry, stable, hydrophobic core. This huddling is actually the primary force that drives a protein to fold into its final functional shape. Without alanine and its hydrophobic cousins, proteins would remain as limp, useless strings, unable to perform the work of life. Now let's look a little deeper into the history and the handedness of this molecule. You may recall from our first lesson that most amino acids come in two versions, a left-handed L form and a right-handed D form. While glycine was symmetrical and lacked a hand, alanine is the very first amino acid where this distinction becomes real. In your body, and in almost every living thing on this planet, we use L-alanine. It is as if life decided eons ago that the entire symphony would be played on left-handed violins. However, there is a fascinating cross-topic connection here. In the world of bacteria, the tiny, ancient organisms that live all around and within us, they sometimes use the forbidden D-alanine to build their cell walls. It's a clever trick. Because our bodies are designed to recognize and break down L-amino acids, the bacteria use the right-handed D form as a kind of molecular armor. It makes their walls resistant to the enzymes that would otherwise dissolve them. It is a reminder that even a tiny change in geometry, the simple flipping of a molecule from left to right, can be the difference between a building block and a shield. Beyond its role as a structural brick, alanine is a tireless worker in your metabolism, acting as a vital bridge between your muscles and your liver. This is known as the glucose-alanine cycle, and it is a beautiful example of how your body manages its resources during activity. Imagine you are out for a long walk, or perhaps working in your garden. Your muscles are burning energy, and as they do, they produce nitrogen as a byproduct. Too much nitrogen in the muscle can be problematic, so the body needs a way to transport it safely to the liver. This is where alanine steps in, acting like a molecular courier. The muscle converts that nitrogen and a bit of pyruvate into alanine. This alanine then travels through your bloodstream, a quiet traveler on a liquid highway, until it reaches the liver. Once there, the liver unpacks the alanine. It takes the nitrogen and prepares it for disposal, and it takes the remaining carbon skeleton and turns it back into glucose or blood sugar. That sugar is then sent back out to the muscles to be used as fuel once more. It is a perfect circular economy of energy and waste management. Alanine isn't just sitting still in a protein wall. It is constantly moving, ensuring that your muscles have the fuel they need and your body stays clean and balanced. As you lie here, your body is likely engaged in this very cycle, quietly moving nutrients and cleaning up the day's work while you drift toward sleep. It is a reassuring thought, isn't it? That even when you are at your most still, there is a steady foundation of molecules like alanine, keeping the lights on and the pathways clear. In the world of biochemistry, we often focus on the star molecules, the complex enzymes that unzip DNA, or the powerful hormones that change our mood. But there is a profound dignity in the ordinary amino acids, like alanine. It doesn't have a fancy ring, and it doesn't form covalent bridges, but its very presence allows the stars to do their jobs. It is the silent partner in every breath you take and every movement you make. It reminds us that consistency is a form of greatness. To be the steady brick, the reliable courier, the hydrophobic anchor. These are the roles that make the complexity of life possible. We have spent our time tonight looking at the strength of the simple methyl group. We have seen how adding just one carbon and 3 hydrogens to our traveler's pack transformed the flexible hinge of glycine into the sturdy foundation of alanine. We have felt the quiet push of the hydrophobic effect, driving the centers of our proteins into a dry, stable sanctuary. And we have followed the courier through the bloodstream, watching as it balances our energy and clears our waste. Let's set that idea down now, fully formed, and let our attention drift toward the more complex shapes that these sturdy bricks can take. We have mastered the single block, the simple CH3 of alanine. But nature rarely stops at a single block. What happens when the scaffolding becomes more elaborate? What happens when the traveler doesn't just carry one brick, but a collection of them, arranged in branching patterns that look like the limbs of a tree? Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about a trio of molecules that act as the heavy timber of your muscles. We are moving from the simple foundation of a single brick to the rugged architecture of the branched chains. We are drifting toward a place where the shapes become more varied, where the V's and the L's of the molecular world provide the strength that allows us to stand tall. But for now, let the steady, reliable image of alanine be your anchor as you drift further into the quiet. Valine, Leucine, and Isoleucine. In our last quiet moment, we spoke of alanine as a simple, sturdy brick, a single methyl group that serves as the humble foundation for so much of our internal architecture. But as we move deeper into the forest of biochemistry, we find that life often requires more than just a flat wall. It requires scaffolding, interlocking joints, and heavy timber to support the immense weight of our biological machines. This brings us to a trio of remarkable travelers known as the branched chain amino acids, or BCAAs. You may have heard their names whispered in the halls of nutrition or seen them on the labels of health supplements, but here, in the quiet of our study, we will see them for what they truly are. The structural anchors of the protein world. These 3 are valine, leucine, and isoleucine. Imagine, if you will, a carpenter building the frame of a great house. While alanine was a single, straight peg, these 3 are like sturdy, 3-dimensional joints. They are called branched chain because their R groups do not move in a single, straight line of carbons. Instead, they fork and divide, creating shapes that look like the letters V or L, or perhaps like the sturdy crotch of a tree branch, where 2 limbs meet. This branching isn't just a visual curiosity. It changes the way these molecules occupy space. Because they are wider and more complex than alanine, they act like heavy timber, providing a rigid, unyielding density to the proteins they inhabit. Let's look at the first of our trio, valine. In your mind's eye, see that central alpha carbon we discussed. Instead of holding just one carbon atom like alanine, valine holds a carbon that immediately splits into 2 branches. This creates a shape that looks exactly like a V. Think of Valine as a sturdy tripod. Because of this shape, it is very difficult to compress. When valine is tucked into a protein, it acts like a structural spacer, ensuring that the folds of the protein don't collapse in on themselves. It provides the elbow room that enzymes need to maintain their precise working shapes. Next, we meet leucine, perhaps the most famous of the three. Leucine is slightly longer than valine, with its branch occurring one step further away from the alpha carbon. It looks like a long, sturdy L. Leucine is the most abundant of the branched chain amino acids in your muscles, and it carries a very special responsibility. It isn't just a building block, it is a signal. In the complex world of your metabolism, leucine acts like a foreman on a construction site. When leucine levels rise in your bloodstream, it flips a switch in your cells, a pathway known as mTOR that tells your body it is time to start building and repairing muscle tissue. It is the molecular go signal for growth. Without leucine, even if you had all the other building blocks ready, the construction crew would stay idle, waiting for the command to begin. The third member of our trio is isoleucine. As its name suggests, it is an isomer of leucine, meaning it contains the exact same atoms, 6 carbons, 13 hydrogens, 1 nitrogen, and 2 oxygens, but they are arranged in a slightly different pattern. If leucine is a straight arm with a hand turned to the side, isoleucine is like an arm with the bend at the elbow. This subtle change in geometry allows isoleucine to fit into different niches within a protein. Interestingly, isoleucine is one of the few amino acids that has a second chiral center, another point of handedness within its side chain. This makes it even more specific in how it can bond and interact with its neighbors. It is a master of precision, ensuring that the structural timber of our bodies is fitted together with the accuracy of a fine watch. We often call these three aliphatic, a word that sounds technical but has a very earthy origin. It comes from the Greek word for fat or oil. This tells us everything we need to know about their personality. Like the alanine we studied, these three are fiercely hydrophobic. They have no interest in the watery world of the cytoplasm. When a protein is being born, these three huddle together with an almost magnetic intensity. Because of their branched shapes, they don't just huddle, they interlock. Imagine trying to pull apart 2 pieces of Velcro or 2 tangled tree branches. That is how valine, leucine, and isoleucine behave in the core of your proteins. They create a hydrophobic knot that is incredibly difficult to untie. This is why the proteins in your muscles can withstand the immense physical tension of lifting, running, and jumping without snapping or losing their shape. There is a poignant example of how important the balance of these 3 molecules is to our health, found in a condition known as maple syrup urine disease. It is a rare genetic struggle where the body lacks the specific enzyme needed to break down the waste of these 3 amino acids. When valine, leucine, and isoleucine cannot be processed, they begin to build up in the blood, and a byproduct of this buildup has a very distinct, sweet smell, exactly like maple syrup. While the name sounds charming, the reality is a stark reminder of the delicate precision of our chemistry. It shows us that every molecule we welcome into our bodies must have a clear path not only for construction, but also for a graceful exit. We are a river of chemistry, and the flow must never be blocked. As you rest tonight, your body is likely managing this very flow. If you moved your body today, if you walked, or lifted, or stretched, your cells are currently using leucine to signal for repairs. They are using valine and isoleucine to reinforce the timber of your muscle fibers. ensuring they are ready for the morning. It is a quiet, tireless renovation project happening in the dark, driven by these branched chain travelers. You are being rebuilt, bit by these tiny V's and L's, while your mind drifts further away from the day's concerns. We have seen the strength of the tripod, the command of the foreman, and the precision of the isomer. We have felt the oily, hydrophobic pull that keeps our internal structures solid and dry. We have come a long way from the single hydrogen of glycine and the single brick of alanine. We are beginning to see how life builds complexity, not by inventing entirely new rules, but by subtly rearranging the same atoms into more and more sophisticated shapes. The scaffolding is now in place. The interior walls are sturdy. The heavy timber is supporting the weight of the biological roof. Let's set that idea down now, fully formed, and let our attention drift toward a different kind of architectural challenge. We have explored the sturdy, straight timbers and the branched joints. But what happens when a structure needs to do more than just stand still? What happens when we need a permanent, rigid bend in our path, a corner that can never be straightened? Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about an amino acid that doesn't follow the rules of a straight line or a simple branch. We are moving toward a rebel, a molecule that reaches back and grabs its own tail, creating a rigid ring that forces life to take a sharp turn. We are headed toward Proline, the architect of the curve, the one who ensures that our proteins have the shape and the kinks they need to function. But for now, just let the strength of the branched chains support you. Feel the stability they provide to your frame, and let that sense of structural peace carry you deeper into the quiet. Proline. In our last quiet session together, we stood among the sturdy timbers of the branched chain amino acids. We felt the strength of the tripod, the command of the foreman, and the precision of the isomer. We saw how Valine, Leucine, and Isoleucine act as the heavy, interlocking joints that provide our muscles with their rugged architecture. We explored the idea of straight lines and branching paths, all moving outward from that central alpha carbon like limbs on a tree. But now, as we continue our gentle journey through the alphabet of life, we encounter a rebel, a molecule that refuses to follow the standard geometry of its cousins. This is Proline, the architect of the curve, the molecule that ensures life never moves in a perfectly straight line. To understand why Proline is such a unique character, we have to look back at the standard structure we learned at the very beginning. Remember that every amino acid has an amino group on one side and a carboxyl group on the other, with a central carbon in the middle holding a side chain. In every other amino acid, that side chain reaches out into the world like an arm. But Proline is different. Proline's side chain reaches out, but then it does something extraordinary. It loops all the way back around and shakes hands with its own amino group. This creates a rigid, five-membered ring that includes the nitrogen atom of the backbone itself. Because of this loop, chemists sometimes refer to Proline not as an amino acid, but as an amino acid. It is the only one of the 20 to have this cyclic structure where the side chain is fused to the nitrogen. Imagine, if you will, a long, soft silk ribbon representing a protein chain. Most amino acids are like small decorative beads strung along that ribbon, allowing it to twist and flow in the wind. But Proline is a heavy industrial staple driven through the ribbon. Once that staple is in place, the ribbon can no longer move freely. It is forced into a sharp, permanent angle. Proline doesn't just suggest a turn, it mandates it. This rigidity makes Proline the great architect of the kink. In the world of protein structure, there is a very common shape called the alpha helix, which looks like a beautiful, winding spiral staircase. Most amino acids love to sit in this spiral, holding onto the step above and the step below. But because of its rigid ring, Proline cannot fit into the staircase. It is too stiff to make the gentle curve. When a proline appears in a sequence, it acts as a helix breaker. It puts a sudden stop to the spiral and forces the protein to go in a completely different direction. This might sound like a disruption, but it is actually a vital piece of engineering. Without these sharp turns, proteins would just be long, straight poles. Proline allows them to fold into compact, intricate globes, the shapes they need to become enzymes, antibodies, and receptors. One of the most profound places we see proline at work is in the protein we touched upon in our study of glycine, collagen. You'll remember that collagen is the triple-stranded rope that provides the strength for your skin, your bones, and your joints. While glycine provides the tiny space needed for the strands to pack together, proline provides the twist. About 1 out of every 4 amino acids in collagen is a proline. These prolines act like the tension in a spring, forcing the strands into that tight helical shape. It is the rigidity of proline that makes your skin elastic and your tendons tough enough to pull against your bones. But there is a deeper layer to this story, a classic example of how a single chemical modification can change the course of history. Within the collagen of your body, proline often undergoes a transformation. An enzyme comes along and adds a hydroxyl group, an oxygen, and a hydrogen atom to the proline ring, turning it into hydroxyproline. This extra little hand allows the prolines to form hydrogen bonds between the strands of the collagen rope, acting like molecular glue that locks the 3 strands together. Collagen molecule. Structure of a collagen fibers. However, the enzyme that creates hydroxyproline has a requirement. It cannot do its job without the presence of vitamin C. This is the secret behind the ancient sailors' struggle with scurvy. When those sailors spent months at sea without fresh fruit, their vitamin C levels would drop. Without vitamin C, their bodies could no longer turn proline into hydroxyproline. The glue in their collagen began to fail. Their old wounds would reopen, their teeth would loosen, and their skin would lose its integrity. It is a stark and gentle reminder of how much we rely on the smallest details of our chemistry. The strength of your entire physical frame depends on a tiny ring of carbons and the specific vitamins that help them hold their shape. As you rest tonight, your body is constantly managing this architecture. Your fibroblasts are busy weaving new collagen fibers, using proline to create the necessary turns and twists that keep your tissues healthy. They are utilizing the rebel molecule to ensure that your internal structures aren't just rigid, but are intelligently designed with the right bends in the right places. Proline teaches us that sometimes the most important thing we can do is stay firm in our position to provide the kink in the chain that allows the rest of the system to fold into something meaningful. Beyond the physical structure, proline also plays a role in how your body responds to stress. It is often found in high concentrations in the proteins that protect plants and animals from freezing or from extreme salt concentrations. Because its ring structure is so stable, it acts as a chaperone, helping other proteins maintain their shape even when the environment becomes harsh. It is a molecule of resilience. It is the part of you that refuses to be pushed out of shape when things get difficult. We have seen how this structural rebel breaks the rules of the straight line. We have felt the tension of the rigid ring and seen how it forces the protein ribbon to turn. We have looked at the historical lesson of the sailor and the vitamin C, seeing how a simple hydroxyl group on a proline ring is the difference between health and decay. We have moved from the flexible hinge of glycine to the sturdy brick of alanine, through the heavy timber of the branched chains, and now through the permanent, rigid bends of Proline. Our molecular landscape is becoming more diverse. We have the anchors, the joints, and the turns. But there is another element we haven't yet explored in our quiet fireside chat, an element that brings a different kind of energy to the chain. So far, we have been building with carbon, hydrogen, and nitrogen, but there are travelers in this group who carry the sun and the fire within them, using an atom that is rarer and more reactive. And with that understanding of shape quietly in place, we're naturally led to ask, what about the elements that bring a bit of fire or sun to the chain? What happens when we introduce sulfur into the mix? Sulfur is a heavy, yellow element, and it brings with it a special kind of reactivity. Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about the amino acid that acts as the start signal for almost every protein your body creates. We are moving toward methionine, the ignition key of the biological world. We are leaving the world of pure structural architecture and entering the world of timing, signals, and the very first steps of creation. But for now, let the rigid, reliable turns of Proline give you a sense of order. Let the strength of your own collagen support you as you drift further into the stillness. Methionine. In our last quiet session, we saw how Proline creates the bends in the road, forcing the silk ribbon of life to take a sharp, permanent turn. But every road, no matter how winding, needs a beginning. Every great machine needs a spark to roar into life. This brings us to a traveler of immense importance, one who carries a secret hidden within its backpack. This is methionine. Up until this point, our molecular journey has been built primarily from carbon, hydrogen, and nitrogen, the standard elements of the organic world. But methionine introduces something rare and potent into the mix, an atom of sulfur. In the ancient language of the earth, sulfur is the element of fire and sun. In the quiet world of your cells, however, sulfur is not a flame, but a glowing coal, tucked away safely within a chain of carbons. If you were to look at the side chain of methionine, you would see a short string of carbons, and then, nestled between them, is that sulfur atom. We call this a thioether bond. Imagine a long, dark bridge over a golden stream. The sulfur is that stream, providing a unique kind of flexibility and a soft electronic glow that makes methionine stand out from its purely carbon-based cousins. Because the sulfur is capped by a final methyl group at the end of the chain, it is nonpolar and hydrophobic. It likes to hide in the interior of proteins, just like alanine or valine, but it brings with it a subtle, reactive potential that those simpler molecules lack. The most extraordinary thing about methionine is its role as the universal start signal. Imagine a vast, silent factory filled with thousands of different machines. Some of these machines make the hemoglobin that carries your oxygen. Others make the insulin that balances your sugar. But none of these machines can begin their work without a specific ignition key. In the grand library of your DNA, every single instruction for a new protein begins with the same three-letter code, A, UG. And that code, always, without exception, calls for methionine. It is the first amino acid added to almost every protein chain as it is being born. Whether you are a blue whale, a redwood tree, or a human child drifting off to sleep, your biology begins its creative work with methionine. It is the molecular once upon a time that starts every story. In bacteria, They use a slightly modified version called N-formyl methionine, which acts as a flag for your immune system to recognize invaders. But in your own cells, it is pure methionine that turns the key. It ensures that the protein-building machinery, the ribosome, is in the exact right place to begin the translation of life. But methionine is much more than just a starting point. It is also the master of the methyl group. You may remember the methyl group from our study of alanine, that small, sturdy block of 1 carbon and 3 hydrogens. Methionine is the body's primary source of these blocks. Through a beautiful and complex series of reactions, methionine is transformed into a molecule called SAM, or S-adenosyl methionine. Think of SAM as a molecular stapler. It travels through the cell, carrying a single methyl group, and staples it onto other molecules, including your DNA itself. This process, called methylation, is the heart of epigenetics. It is the way your body tags certain genes to be turned off or on. It's as if methionine provides the sticky notes that help your cells remember which instructions to follow and which to ignore. This tiny traveler, by donating its methyl group, helps regulate everything from your mood to your metabolism. It is a quiet, powerful influence on the very expression of your identity. To keep this vital ignition and tagging system running, your body relies on a delicate balance of vitamins, specifically vitamin B12 and folate. These vitamins work together in a rhythmic cycle to recycle methionine, ensuring that once it has given away its methyl group, it can be rebuilt and ready to work again. It is a perfect example of the circular economy of the cell. Nothing is wasted, and every spark is preserved to be used once more. Because methionine is an essential amino acid, we cannot manufacture it ourselves. We must find it in the world around us. It is found in abundance in things like eggs, sesame seeds, and Brazil nuts. When you eat these foods, your body isn't just taking in calories. It is gathering the ignition keys it needs for tomorrow's growth and the methyl tags it needs to manage your genetic library. It is a profound realization that the food you ate today provides the very first link in the proteins your body will build while you sleep tonight. As you lie here, your body is likely in the middle of a grand recycling project. The methionine you consumed is being activated. Its sulfur, coal, is glowing with potential, and its methyl groups are being prepared to tag your DNA, ensuring your cells are functioning exactly as they should. It is a process of incredible precision, happening millions of times a second in the quiet dark of your internal world. Methionine teaches us the importance of the beginning. It reminds us that every complex structure, every long-form protein, and every biological process must have a clear and consistent starting point. It shows us that one single, unique element, the sulfur, can change the entire function of a molecule, transforming a simple building block into a master key. We have explored the thioether bridge and the golden stream of sulfur. We have watched the start signal being given in the cellular factory. We have seen how the methyl tags are stapled onto the DNA, guiding the expression of life itself. We have traveled from the structural turns of proline to the ignition spark of methionine. But our story of sulfur is only half told. In methionine, the sulfur is hidden, tucked away in the middle of the chain like a secret. But what happens when that sulfur is exposed? What happens when it is no longer a capped coal, but a bare, reaching hand? So what happens when sulfur isn't just a hidden coal, but a hand reaching out to grab another? That's the question we'll gently explore next, as we meet methionine's cousin, cysteine. We will see how an exposed sulfur atom creates the strongest handshakes in the molecular world, pinning our proteins into their final, enduring shapes. But for now, let the steady glow of methionine guide your thoughts. Feel the quiet power of your body's ignition system, ready to begin anew with every breath as you drift further into the stillness. Cysteine. We just felt the heat of the sulfur in methionine, tucked away like a glowing coal in a protective hearth of carbons. But its cousin, cysteine, is a far more extroverted traveler. In cysteine, that sulfur atom is not hidden or capped. It is exposed, sitting right at the tip of the side chain, bonded only to a single hydrogen atom. We call this group a thiol or a sulfhydryl group, written in our chemical shorthand as SH. Imagine cysteine like a person walking through a crowded room with their hand held out, searching for a partner. While many other amino acids are content to simply nudge against one another or hide away from water, cysteine is looking for a commitment. When two cysteines find each other within a folding protein chain, they reach out and perform what we call a molecular handshake. The two sulfur atoms drop their hydrogens and bond directly to each other, forming a disulfide bridge, RSSR. This isn't just a casual interaction, it is a covalent bond, one of the strongest and most permanent connections in all of biology. Think of it like a molecular safety pin. If the protein is a long, winding ribbon of silk, the disulfide bridges are the silver pins that lock the folds into place, ensuring that no matter how much the ribbon is tossed or turned, it maintains its specific, functional shape. Cysteine is the bridge builder of the microscopic world, the one who ensures that once a protein finds its purpose, it has the structural integrity to stay that way. This strength is why cysteine is the star of one of the toughest materials your body produces, keratin. Keratin is the primary protein in your hair, your fingernails, and the outer layer of your skin. It is also what makes up the claws of a lion, the feathers of a bird, and the horns of a rhinoceros. The reason these structures are so durable is that they are packed with cysteines, all locking hands in a dense forest of disulfide bridges. We can see a beautiful, real-world example of this chemistry in the way we style our hair. If you have ever had a permanent wave or a chemical straightening treatment, you have participated in a high-stakes laboratory experiment. A hairstylist applies a reducing agent to break those sulfur handshakes, making the hair soft and moldable. Then, they shape the hair into curls or pull it straight and apply an oxidizing agent to force the cysteines to lock hands again in their new positions. The perm is permanent because you have literally re-engineered the architecture of your proteins, forcing the molecular safety pins into a new configuration. It is a reminder that the textures of our physical world, the softness of a curl, or the hardness of a nail, are simply the outward expression of millions of tiny sulfur atoms choosing where to hold on. But cysteine's work extends far beyond just structural support. It is also a vital protector of your internal environment. Cysteine is the key ingredient in glutathione, which scientists often call the master antioxidant. Imagine your cells as a busy city. Like any city, the process of living and producing energy creates waste, specifically reactive molecules called free radicals that can damage your DNA and your membranes. Glutathione is the city's elite cleaning crew. Because of that reactive sulfur atom, it can mop up these dangerous free radicals, neutralizing them before they can cause harm. Without a steady supply of cysteine, your body's ability to create glutathione would falter, leaving your cells vulnerable to the rust of oxidative stress. It is a molecule of incredible self-sacrifice. It uses its own reactivity to shield the rest of your biology from the fires of metabolism. It teaches us that being reactive isn't a flaw when directed properly. It is the very thing that allows for protection and enduring strength. Even the way we perceive flavor is touched by this molecule. Have you ever noticed the distinct, savory aroma of roasting meat or the pungent scent of onions and garlic? These are often the scents of sulfur compounds being released and transformed. Cysteine is a major contributor to the Maillard reaction, the complex chemical dance between amino acids and sugars that gives browned food its delicious smell and taste. It seems that whether it is building a bridge, protecting a cell, or flavoring a meal, cysteine is always right in the middle of the action, reaching out to change the world around it. As you drift closer to sleep, consider the quiet strength of the handshakes happening within you right now. Your hair is held in its shape by millions of these tiny silver pins. Your cells are being swept clean of the day's debris by the sulfur-rich guardians of your immune system. Your skin is being reinforced by the bridge builders that ensure you remain a cohesive, protected whole. You are a masterpiece of structural engineering, held together by the quiet, tireless commitment of cysteine. There is a profound sense of security in knowing that your biology doesn't just rely on gravity or chance to hold its shape. It relies on specific, intentional bonds, the covalent handshakes that refuse to let go. Cysteine reminds us that for life to be complex, it must be stable. And for it to be stable, it must have members willing to lock hands and stand firm. We have felt the strength of the bridge and the protection of the antioxidant. We have seen how the exposed sulfur atom of cysteine transforms a simple traveler into a master of stability. We have moved from the hidden coals of methionine to the open handshakes of the bridge builder. Our molecular landscape is now reinforced and pinned into place. But life is not made solely of bridges and anchors. There is also a need for depth, for bulk, and for a certain kind of aromatic stability that comes from the largest, heaviest members of our family. Let's set that idea down now, fully formed, and let our attention drift toward the heavier, more fragrant parts of our chemistry. We are leaving the world of reactive sulfurs and entering a realm of great, flat rings and structures that don't reach out to shake hands, but instead provide a deep, resonant stability to the core of our proteins. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed about a trio of molecules that carry the scent of the ancient earth within them. We are moving toward the aromatics, beginning with phenylalanine. We are headed toward the world of heavy rings and hidden depths, where the architecture is no longer about pins and handshakes, but about the solid, unyielding bulk of the aromatic core. But for now, let the safety pins of cysteine keep you secure. Feel the strength of your own internal bridges, and let that sense of structural peace carry you deeper into the quiet of the night. Phenylalanine. In our last quiet moment, we spoke of the strong bonds of cysteine, those silver safety pins that lock the folds of life into their permanent, enduring shapes. We felt the security of the disulfide bridge, the structural commitment of the sulfur handshake. But as we move further into our rhythmic biochemistry deep dive, we leave the world of reactive hands and enter a realm of great, stable depth. We move into the world of the aromatics. This is a group of amino acids that carry a specific kind of molecular scent, not a smell you would detect with your nose, but a fragrance of stability that defines the very core of our proteins. Let us focus our attention on the first of these heavyweights, phenylalanine. To understand phenylalanine, we should look back at the very first brick we placed in our foundation, alanine. You remember alanine as a simple, sturdy carbon with its 3 hydrogen companions. Now, imagine taking that alanine and, instead of leaving it as a small block, we attach a massive, flat, shimmering ring to it. This is the phenyl ring, a 6-carbon hexagon where electrons dance in a continuous, rhythmic circle. In the language of chemistry, this is a benzene ring. When we join the two, we get phenylalanine, a name that literally tells its own story, a phenyl group joined to an alanine. Think of phenylalanine as a large, flat stone skipped across a pond. Because of that big, flat ring, it is incredibly bulky. It takes up a significant amount of space, but more importantly, it is fiercely hydrophobic. Just like the skipping stone stays on top of the water because of its shape and momentum, phenylalanine refuses to mix with the watery environment of the cell. While the smaller molecules huddle, phenylalanine stacks. Because these rings are flat, they like to lay on top of one another like a stack of plates, creating a dense, unyielding core within a protein. If a protein were a grand cathedral, phenylalanine would be the heavy, unmovable foundation stones buried deep beneath the floor, providing the bulk and weight that prevents the structure from ever shifting. This bulkiness is why phenylalanine is so vital for the stability of enzymes. Many of the machines in your body need a very specific pocket to catch their target molecules. Phenylalanine often acts as the stuffing or the padding that creates the walls of these pockets. It is reliable, it is heavy, and it doesn't react with things easily. It provides a solid, non-polar environment where chemical reactions can take place without interference from the outside world. There is a deeper layer to this molecule, one that brings us to a significant figure in the history of medicine, a Norwegian doctor named Asbjørn Felling. In the 1930s, Dr. Felling was approached by a mother whose 2 children struggled with severe intellectual disabilities and a strange, musty odor. Through patient observation and careful chemistry, he discovered that these children could not process phenylalanine. This was the discovery of PKU, or phenylketonuria. Normally, your body takes the phenylalanine you eat and converts it into another amino acid called tyrosine using a special enzyme. But in those with PKU, that enzyme is missing. The phenylalanine builds up in the blood like a river that has been dammed, eventually spilling over into the brain where it can cause damage. This discovery was a turning point in human history. It led to the very first newborn screening tests. Today, every baby born in a modern hospital receives a small heel prick to check for this balance. It teaches us a profound lesson that even the most stable, foundational building block must be kept in perfect harmony. Life is not just about having the pieces. It is about the graceful flow of one piece into the next. We see a very practical, real-world connection to this in our daily lives, often found on the back of a soda can or a packet of sugar substitute. If you have ever looked at the warning label on a product containing aspartame, you will see the words, phenylketonurics, contains phenylalanine. Aspartame is a dipeptide, a tiny chain of 2 amino acids, and one of them is phenylalanine. Because phenylalanine is so stable and can interact with the sweet receptors on your tongue with such precision, it provides that intense sweetness without the calories of sugar. For most of us, it is a simple convenience. But for those with PKU, it is a reminder of the delicate chemical balance they must maintain every day. As you lie here, your body is managing this balance with effortless grace. It is taking the proteins from your dinner, breaking them down into these heavy, aromatic rings, and deciding exactly how many should be used to build your enzymes and how many should be converted into other messengers. It is a quiet, steady sorting process, a heavy-duty architecture happening in the silence of your cells. There is a sense of peace in the bulk of phenylalanine. It isn't a molecule of sudden sparks or rapid changes. It is a molecule of endurance. It is the weight that holds you together, the flat stones that stack to form the heart of your biological machines. It reminds us that for life to be vibrant and active, it must also have a place of stillness and unyielding weight. We have felt the density of the benzene ring and the stacking strength of the aromatic core. We have followed the history of Dr. Falling in the lesson of PKU, seeing how the flow of molecules is as important as their structure. We have moved from the reactive sulfur handshakes of cysteine to the solid, unmoving stones of phenylalanine. Our molecular world is now heavy, stable, and deep. But as we have seen time and again in our journey, nature is never satisfied with just one form. It loves to take a perfect structure and add a tiny, transformative detail. Imagine our large, flat, skipping stone once more, that heavy ring of phenylalanine. What would happen if we were to take that stone and add just one single breath of oxygen to it? What if we attached a tiny hydroxyl group to that massive ring? Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about a molecule that looks almost exactly like phenylalanine, but with one tiny, life-changing addition. We are moving toward tyrosine, the great messenger and precursor of joy. We are moving from the foundation stone to the signal fire, seeing how a single atom of oxygen can turn a structural anchor into a herald of your emotions. But for now, let the heavy, aromatic core of phenylalanine give you a sense of solid ground. Feel the stability it provides to your very existence, and let that weight carry you deeper into the quiet. Tyrosine. We ended our last thought with the heavy ring of phenylalanine, that massive skipping stone of a molecule that provides the unyielding weight to the core of your proteins. It is a structure of immense stability, a silent anchor. But now, imagine taking that stone and adding just one single breath of oxygen to it. Imagine attaching a tiny hydroxyl group, an oxygen atom bonded to a hydrogen to that massive benzene ring. This small addition, just a few atoms in size, transforms the structural anchor into something entirely new. It becomes tyrosine. In the language of biochemistry, adding that hydroxyl group changes the benzene ring into a phenol group. While phenylalanine was content to huddle in the dark, dry interior of a protein, tyrosine is more adventurous. That little oxygen atom acts like a molecular handle. If phenylalanine is a smooth, locked door that you can only lean against for support, tyrosine is a door with a polished brass handle that can be turned, pulled, and used to communicate with the world outside. This handle allows tyrosine to participate in hydrogen bonding, making it far more versatile than its purely hydrophobic cousin. It can sit at the boundary where the protein meets the water, reaching out to touch the liquid environment while keeping its heavy ring firmly planted in the protein's heart. To understand the true power of this molecular handle, we must look at a process that is fundamental to every thought and movement you possess: phosphorylation. In the landscape of your cells, information is passed along not just through electrical wires, but through a series of chemical handshakes. A special kind of enzyme, called a tyrosine kinase, takes a high-energy phosphate group and staples it onto the hydroxyl handle of a tyrosine molecule within a protein. Think of this like a molecular light switch. When the tyrosine is naked, the protein is in its off state. But once the phosphate is attached, the switch is flipped on. This change in shape and charge acts as a signal to the rest of the cell, telling it to grow, to divide, or to take in nutrients. This is the basis of receptor tyrosine kinases, or RTKs, which sit on the surface of your cells like sophisticated satellite dishes, waiting for a signal from the outside, perhaps a growth factor or insulin, to tell them it is time to act. When you feel your body responding to the food you've eaten or the air you're breathing, you are feeling the rhythmic clicking of these tyrosine switches. It is a system of incredible precision. If the switch gets stuck in the on position, it can lead to uncontrolled growth, a reminder of how vital it is that our biological signals remain as balanced and rhythmic as a slow breath. But the story of tyrosine deepens as we move from the structure of proteins to the creation of the messengers that define your internal weather. Tyrosine is the raw material, the mother molecule, for a family of chemicals called the catecholamines. Your body takes that phenol ring and, through a series of careful steps, transforms it into dopamine, the chemical of reward, motivation, and the simple joy of discovery. It is the feeling of satisfaction when you complete a task, or the anticipation of a pleasant dream. From dopamine, the path continues toward norepinephrine and epinephrine, more commonly known as adrenaline. These are the heralds of excitement and action. They are the chemicals that make your heart race and your mind sharpen when you encounter a challenge. It is a profound thought to realize that the same heavy ring that provides stability to your skin and bone is also the source of your most intense emotions. Your joy, your focus, and your courage are all literally built from the same aromatic foundation. We are, in a very real sense, walking symphonies played on the strings of tyrosine. Beyond the brain, tyrosine travels to the base of your throat, where it meets the thyroid gland. Here, tyrosine performs one of its most unique tasks. It pairs up with atoms of iodine, one of the few places in your body where this heavy element is used to create the thyroid hormones, $T, $3, and $T, $4. These hormones are the master regulators of your metabolism. They determine how fast your cells burn fuel and how much heat your body produces. If tyrosine is the handle on the door of the cell, in the thyroid, it becomes the thermostat for your entire existence. It ensures that the fires of your biology burn at a steady, gentle glow, neither too hot nor too cold, allowing you to rest in perfect thermal harmony. Even the way you present yourself to the world is a gift from tyrosine. Within your skin and hair, an enzyme called tyrosinase takes this amino acid and weaves it into melanin, the pigment that gives us our beautiful variety of colors. Melanin isn't just about appearance. It is a shield, a complex polymer that absorbs the energy of the sun and protects your DNA from damage. Whether you are pale or deep-toned, your protection and your color are written in the ink of tyrosine. It is the molecule that wears our history and our protection on its sleeve. As you lie here, drifting toward the threshold of sleep, your body is quietly managing all these roles. It is using tyrosine to synthesize the dopamine that will help your brain process the day's memories. It is maintaining the steady rhythm of your thyroid to keep you warm through the night. It is ensuring that your cellular switches are set to rest and repair. You are a masterpiece of signal and response, a long-form narrative written in aromatic rings and hydroxyl handles. There is a reassuring depth to tyrosine. It teaches us that stability, the benzene ring, and reactivity, the hydroxyl group, are not opposites, but partners. It shows us that from a solid foundation, we can build the most delicate signals of joy and the most powerful hormones of action. It reminds us that even the smallest change, the addition of a single oxygen atom, can open a door to an entirely new world of possibility. We have felt the turn of the molecular handle and the rhythmic click of the phosphorylation switch. We have traced the path from the heavy stone of the ring to the signal fires of dopamine and adrenaline. We have seen how tyrosine wears our colors and regulates our warmth. We have moved from the silent, structural anchor of phenylalanine to the vibrant, communicative herald of tyrosine. Our molecular landscape is now alive with signals, messengers, and pigments. In our last quiet session together, we ended with the idea of tyrosine as a precursor to joy. Let's pick up that thread right where we left it and look at the largest traveler of them all, We have seen what a single ring and a single handle can do, but what happens when the architecture becomes even more grand? What happens when we look at a molecule that doesn't just hold a handle, but possesses a massive double-ring structure that guards the very gates of our sleep? Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed about the amino acid that is likely helping you drift away even now. We are moving toward tryptophan, the guardian of your rest and the largest of all the building blocks. We are headed toward the world of the indole ring, where the fragrance of the aromatics turns toward the deep velvet curtains of the night. But for now, let the signal fires of tyrosine burn low and steady. Feel the warmth of your own metabolism and the quiet joy of understanding, and let that sense of peace carry you deeper into the quiet. Tryptophan. In our last quiet session together, we ended with the idea of tyrosine as a precursor to joy. Let's pick up that thread right where we left it and look at the largest traveler of them all. We have seen what a single ring and a single handle can do, but now the architecture of life becomes even more grand, even more imposing. If tyrosine and phenylalanine were single-room cottages of stability, we are now approaching a cathedral. This is tryptophan, the guardian of your rest and the most substantial of the 20 building blocks. In the landscape of the cell, tryptophan is easily recognized by its massive double-ring structure, known as an indole ring. Imagine, if you will, the 6-carbon benzene ring we studied in phenylalanine. Now, imagine a second, 5-membered ring containing a nitrogen atom fused directly to its side. This is not just a molecule, it is a heavy, ornate gate. In the world of biochemistry, tryptophan is the giant among the travelers. Because it is so large and carries so many atoms, it is the rarest amino acid found in your proteins. It is as if the cell knows that such a massive structure must be used sparingly, placed only where its unique weight and shape are truly required. To understand tryptophan, we must first recognize its essential nature. While your body is a master chemist, capable of weaving many molecules from scratch, it lacks the blueprints to build the complex indole ring of tryptophan. It is simply too expensive for your cells to manufacture. Instead, we rely on the plants and the world around us to do the heavy lifting. They capture the energy of the sun and the nitrogen of the soil to forge these double rings for us. When you eat, you are essentially importing these ready-made velvet curtains to hang within your own biological house. It is a beautiful, humble connection between your rest and the green world outside your window. You may have heard tryptophan mentioned in the context of a heavy meal, perhaps during a holiday feast. There is a common myth that the tryptophan in turkey is what makes us sleepy after a large dinner. The truth, as is often the case in biochemistry, is a bit more subtle and far more interesting. While turkey does contain tryptophan, it isn't the amount of the amino acid itself that brings on the slumber. Instead, it is the ratio. Imagine a crowded bus, the large neutral amino acid bus heading toward your brain. Tryptophan is a large, slow passenger, and it has to compete with many other amino acids for a seat on the transport protein that crosses the blood-brain barrier. When you eat a large meal with plenty of carbohydrates, your body releases insulin. This insulin acts like a polite usher, clearing the bus of most other amino acids and sending them to your muscles. This leaves tryptophan as the only passenger left with a seat. It finally has a clear path to enter the brain. Once it crosses that threshold, your biology begins a 2-step transformation that defines your internal rhythm. The first step is the creation of serotonin. In the quiet chemistry of your neurons, tryptophan is converted into this famous neurotransmitter. We often call serotonin the feel-good chemical, but it is more accurately described as the chemical of enoughness. It is the sense of satisfaction, the stabilization of mood, and the quiet confidence that all is well. It is the steady hum of a well-tuned engine. Serotonin doesn't provide the high of dopamine. Instead, it provides the floor. It is the emotional ground upon which you stand. But as the light of day begins to fade and the world turns toward the indigo of dusk, your brain performs the second part of the miracle. Within the pineal gland, that tiny pine cone-shaped structure at the center of your brain, serotonin is transformed once again. It becomes melatonin. If tryptophan is the heavy velvet of the curtain, melatonin is the act of drawing that curtain closed. It is the universal signal to every cell in your body that the sun has set and it is time for the work of repair and restoration to begin. Melatonin doesn't force you to sleep like a drug. Instead, it whispers to your biology that the window of rest is open. It synchronizes your heart rate, your temperature, and your thoughts with the rotation of the earth itself. Beyond the world of sleep and mood, tryptophan has another vital, though often overlooked, role. It acts as a backup generator for your nutrition. If your diet is low in niacin or vitamin B3, your body can actually use tryptophan to manufacture its own. It takes about 60 molecules of tryptophan to make a single molecule of niacin, a conversion rate that shows just how precious this amino acid is. It is a safety net, ensuring that even if your external resources are lean, your internal chemistry has a way to keep the lights on. It reminds us that, in the world of the cell, nothing is ever truly wasted, and every molecule has a depth of purpose that extends far beyond its primary job. In our study of the aromatics, we have seen how life uses rings to create stability and signals. Phenylalanine gave us the heavy foundation stone. Tyrosine gave us the molecular handle and the signal fires of joy. And now, tryptophan gives us the massive, double-ringed guardian of our cycles. Together, these three make up the aromatic core of our biochemistry, the heavy, stable, and deeply influential members of the 20. They are the molecules that interact with light, that absorb energy, and that bridge the gap between our physical structure and our emotional experience. As you lie here now, the tryptophan you have gathered from your day is likely completing its journey. It has crossed the barrier. It has been woven into serotonin to stabilize your mind, and it is now being drawn into the gentle embrace of melatonin. The velvet curtains are being pulled shut across the windows of your consciousness. You are participating in a cycle as old as life itself, a rhythmic transition from action to rest, facilitated by the largest and most complex building block we have. There is a profound sense of security in the size of tryptophan. It is a molecule that cannot be rushed. Because of its bulk and its complexity, its effects are slow, steady, and enduring. It teaches us that the most important transitions in life, the transition from wakefulness to sleep, from anxiety to peace, from hunger to satisfaction, require a certain weight, a certain gravity. We cannot simply switch into sleep. We must be guided there by the heavy indole rings that guard our rest. We have felt the weight of the double ring and the enoughness of serotonin. We have watched the transformation into the melatonin of the night. We have followed the rhythmic bus across the blood-brain barrier and seen the safety net of the niacin connection. We have moved from the single handle of tyrosine to the massive gates of tryptophan. Our molecular landscape is now fully established, with its heavy anchors and its vital signal precursors. We have mastered the world of the nonpolar, the hydrophobic, and the aromatic, the molecules that define the interior, the dark and the structural depths of our proteins. And with that understanding of the heavy rings quietly in place, we're naturally led to ask, what about the molecules that prefer the water? The ones that want to reach out and touch the liquid world. We have spent much of our time in the dry, hidden centers of the protein, but life happens at the edges. Life happens where the protein meets the sea of the cell. Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about a pair of amino acids that are the light-fingered craftsmen of the biological world. We are moving towards serine and threonine, the travelers who carry a tiny drop of water, a hydroxyl group, not as a heavy handle, but as a piece of molecular Velcro. We are leaving the world of heavy aromatic rings and entering the world of the polar and the uncharged, where the molecules are small, agile, and ready to interact with the world around them. But for now, let the heavy, velvet curtains of tryptophan remain closed. Feel the deep, stable rest they provide to your mind, and let that sense of biological harmony carry you deeper into the quiet of the night. Serine and Threonine. In our last quiet moment, we spoke of the heavy, sleepy rings of tryptophan, those velvet curtains that draw across the mind to signal the end of the day's work. We felt the weight of the aromatics, those bulky molecules that huddle in the dry, dark centers of proteins to provide stability and depth. But as we continue our journey, we must open those curtains and step out of the shadows. We are moving from the hidden, oil-loving interior toward the vibrant, liquid exterior of the protein. We are leaving the world of the nonpolar and entering the world of the polar but uncharged travelers. These are the light-fingered craftsmen of the biological world, and their names are serine and threonine. Imagine, if you will, that the protein we have been building is like a grand ship sailing through a vast, salty sea, the cellular fluid. The amino acids we have studied so far, like alanine and leucine, are the heavy timbers and iron bolts kept deep in the hull, away from the corrosive waves. But serine and threonine are the sailors on the deck. They are perfectly comfortable in the spray of the water. In fact, they reach out to touch it. They are the molecular diplomats who mediate the relationship between the solid protein and the watery world around it. The secret to their water-loving nature is a small but powerful addition to their structure, the hydroxyl group. You might remember this from our look at tyrosine. It is a single oxygen atom bonded to a single hydrogen atom, written as OH. While tyrosine carried this group on a massive, heavy ring, serine and threonine carry it on small, agile frames. Let us look first at serine. If you remember alanine, our simple, sturdy brick with a single methyl group, CH3, serine is almost its identical twin. The only difference is that one of the hydrogens in that methyl group has been replaced by an oxygen atom. This tiny change is profound. Suddenly, the brick is no longer just a structural filler. It becomes polar. That oxygen atom is hungry for electrons. It pulls them toward itself, creating a tiny tug-of-war that results in a partial electric charge. This allows serine to form hydrogen bonds with water and with other polar molecules. Think of this hydroxyl group as a piece of molecular Velcro. It is sticky. It wants to grab onto things. Because serine is so small and possessive of this sticky handle, it is often found on the surface of proteins, reaching out into the cellular sea to help the protein dissolve and stay afloat. It is the social amino acid, always ready to form a temporary connection, a soft handshake with the liquid environment. Now, Notice the subtlety of its partner, threonine. If serine is the twin of alanine, threonine is the cousin of valine. It is a bit more complex. It has a branching structure where a carbon holds both a hydroxyl group and another methyl group. This makes threonine a half-and-half molecule. It has a sticky, water-loving side and a dry, oil-loving side. This unique balance allows threonine to act as a bridge builder between the interior and the exterior of a protein. Like isoleucine, threonine also has a second chiral center, a second point of handedness. It is a molecule of extreme precision, designed to fit into very specific niches, where a simple Velcro strip isn't enough, but a full-sized structural timber would be too much. But the true genius of serine and threonine lies in their role as the switches of the cell. Earlier, we whispered about how tyrosine could be phosphorylated, how a phosphate group could be stapled onto it to act as a signal. While tyrosine does this for big, external signals, serine and threonine do it for almost everything else. They are the primary sites for phosphorylation in your body. Imagine a vast control room filled with thousands of toggle switches. Most of those switches are serine or threonine residues. When your body needs to speed up your metabolism, activate an enzyme, or store a memory, a specialized protein called a kinase flies over and drops a heavy, negatively charged phosphate group onto the hydroxyl Velcro of a serine or threonine. This sudden addition of weight and charge forces the entire protein to change its shape, instantly flipping it from off to on. This is how your cells communicate in real time. It is the rhythmic clicking of these millions of tiny switches that allows you to respond to a thought or a heartbeat. You are, in many ways, a beautifully complex circuit board, and serine and threonine are the buttons that keep the electricity of life flowing in the right directions. Beyond their role as switches, serine and threonine are also surgeons. There is a famous family of enzymes called the serine proteases, which includes chymotrypsin and trypsin, the very enzymes that help you digest the proteins you eat. At the heart of these enzymes is something we call the catalytic triad, and serine is the star of the show. Because that oxygen atom in its side chain is so reactive, it can actually perform chemical surgery. It reaches out and physically cuts the peptide bonds of other proteins, breaking them down into their individual amino acids. It is a sharp, precise tool that your body uses to recycle the world around you. We see a beautiful real-world connection to this chemistry in the very fabric of silk. The primary protein in silk is called fibroin, and it is incredibly rich in serine. In fact, the word serine comes from the Latin sericum, meaning silk. It is the presence of these tiny polar travelers that gives silk its unique properties, its incredible smoothness, its soft luster, and its ability to absorb moisture. When you touch a piece of silk, you are feeling the sticky Velcro of millions of serine molecules, reaching out to the world with their gentle, hydroxyl hands. As you lie here, your body is a hive of activity for these hydroxyl workers. Your digestive enzymes are using serine to break down your dinner. Your brain is using phosphorylation to staple the day's lessons into your long-term memory, flipping the serine and threonine switches in your neurons. Your very cells are staying hydrated and balanced because these polar diplomats are mediating the boundary between your solid structure and the liquid life within you. It is a quiet, tireless craftsmanship happening in every corner of your being while you drift towards sleep. Serine and Threonine teach us that being small and sticky is a form of power. They show us that life doesn't just happen in the grand structural timbers or the heavy signal fires. It happens in the subtle, rhythmic flipping of switches and the precise cutting of bonds. They remind us that to be functional, we must be able to interact with the world around us, to have the Velcro that allows us to connect, to change, and to adapt. We have felt the stickiness of the hydroxyl group and watched the flickering of the phosphorylation switches. We have seen the surgeon's precision in the catalytic triad and felt the smoothness of silk in the serine-rich fibers. We have moved from the dry, aromatic curtains of tryptophan to the spray of the cellular sea with serine and threonine. Our molecular landscape is now becoming more reactive, more communicative, and more agile. But as we have seen, nature is always looking to increase the energy. We have explored the molecules that are polar but uncharged, the ones that have a slight tug but don't carry a full electric load. What happens when that tug becomes a full pull? What happens when the side chain doesn't just have a sticky handle, but carries a permanent, buzzing electric charge? So what happens when these side chains become even more reactive, perhaps even carrying a charge of their own? That's the question we'll gently explore next. As we meet the powerhouses of the amino acid family, we are moving toward the acids, the molecules that carry the negative sparks of the biological world. We are headed toward aspartic acid and glutamic acid, the ones who drive our thoughts and our chemistry with the power of electricity. But for now, let the light-fingered craftsmen, Serine and Threonine, continue their quiet work. Feel the balance they bring to your hydration and your memory, and let that sense of intricate harmony carry you deeper into the quiet of the night. Aspartic acid and glutamic acid. In our last quiet session, we felt the light, sticky Velcro of the hydroxyl workers, Serine and Threonine. We watched as they mediated the boundary between the solid protein and the liquid world, flipping the switches of our memories and our metabolism with the rhythmic click of phosphorylation. But as we move forward, the energy of our journey begins to hum with a different kind of intensity. We have seen the structural anchors, the flexible hinges, and the sticky craftsmen. Now, we meet the powerhouses. We meet the molecules that do not just reach out to touch the world, but pull on it with the force of electricity. These are the acidic amino acids, aspartic acid and glutamic acid. To understand these two, we must first look at their names. In the bustling, watery world of your cells, these molecules almost never exist as acids in the traditional sense. Because the environment of your body is slightly alkaline, these amino acids do what all good acids do. They give away a piece of themselves. They release a proton, a single hydrogen ion, into the surrounding fluid. Once that proton is gone, the side chain is left with a permanent, buzzing, negative charge. This is why scientists often call them aspartate and glutamate. They are no longer neutral travelers. They are ions. They are tiny molecular magnets, crackling with an invisible pull that dictates the very shape and function of your biology. Let us look at their shapes, for in biochemistry, shape is destiny. Imagine the central alpha carbon we have studied so faithfully. Aspartate is relatively short. Its side chain is a 2-carbon bridge ending in a carboxyl group. Glutamate is its slightly larger sibling, possessing a 3-carbon bridge. Think of them as 2 different sizes of electric coils. Because they carry a negative charge, They are hydrophilic in the extreme. They don't just like water, they crave it. You will almost always find aspartate and glutamate on the very outermost surface of a protein, reaching into the cellular sea, or tucked into active sites, where they can use their electric pull to grab onto positively charged molecules. Imagine a protein is like a complex piece of origami. Most of the folds are held together by the gentle huddling of the hydrophobic amino acids, or the handshakes of cysteine. But aspartate and glutamate provide a different kind of bond, the salt bridge. When one of these negatively charged acids finds a positively charged partner, like lysine or arginine, they snap together with the force of magnetic attraction. This is one of the strongest ways a protein can maintain its three-dimensional structure. It is the bolt that reinforces the glue. These electric sparks ensure that your enzymes don't just drift apart when the temperature rises or the environment changes. They are the guardians of structural integrity. But the true genius of these two, particularly glutamate, lies in the way they drive the very engine of your consciousness. In the vast, sparking forest of your brain, glutamate is the king. It is the primary excitatory neurotransmitter. If your brain were a grand orchestra, glutamate would be the conductor's baton, signaling for more volume, more speed, and more intensity. When one neuron wants to send a signal to another, it releases a cloud of glutamate into the tiny gap between them, the synapse. This glutamate acts like a key, fitting into specific receptors that allow positive ions to flood into the next neuron, causing it to fire. This is the basis of everything you think, feel, and learn. We call this neuroplasticity. When you learn a new skill, perhaps the very biochemistry we are discussing now, your brain strengthens the connections between certain neurons by using glutamate. It is the gas pedal of your mind. Without the negative charge of glutamate, the electrical symphony of your thoughts would fall silent. It is a profound realization that the same molecule that helps build your muscle fibers also carries the spark of your curiosity and the weight of your memories. It reminds us that in the world of the cell, a single tool can have 1,000 different purposes. There is a beautiful, savory connection to this molecule that you have likely experienced at your own dinner table. Have you ever tasted something so delicious, so deeply satisfying, that it felt like a full flavor? This is umami, the fifth basic taste. In 1908, a Japanese chemist named Kikunai Ikeda discovered that this specific savoriness comes from the L-glutamate molecule found in seaweed, tomatoes, and aged cheeses. He eventually created MSG, or monosodium glutamate, to bring that savory spark to food everywhere. When you eat something rich in glutamate, the receptors on your tongue are literally detecting the presence of this amino acid. Your body interprets this as a signal that you are eating high-quality protein, sparking a sense of reward and satisfaction. It is a bridge between the laboratory and the kitchen, a reminder that the powerhouses of our chemistry are also the sources of our simplest culinary pleasures. Whether it is the depth of a slow-cooked broth or the intensity of a sharp Parmesan, you are tasting the electric pull of the acidic side chain. However, as with all powerful things, balance is essential. Because glutamate is so effective at making neurons fire, your body must be incredibly careful about how much is present at any given time. If too much glutamate lingers in the gaps between your brain cells, the neurons can become overexcited and damage a process known as excitotoxicity. To prevent this, Your brain employs a massive fleet of cleanup cells that vacuum up excess glutamate the moment its job is done. It is a rhythmic, constant process of activation and restraint. It teaches us that the gas pedal is only useful if you also have a way to let off the pressure. Your biology is a master of this ebb and flow, ensuring that the electric sparks of your thoughts remain a gentle light rather than a consuming fire. As you lie here, your brain is likely performing this very cleanup. The glutamate that drove your thoughts throughout the day is being reclaimed and recycled, allowing your neurons to settle into the quiet of the night. Your proteins are being held together by the silent, magnetic pull of the salt bridges, ensuring your body remains a cohesive whole while you rest. The powerhouses are still active, but they are working in the background, maintaining the steady, low-voltage hum of a body at peace. We have felt the electric pull of the negative charge and watched the conductor's baton of glutamate drive the symphony of the mind. We have tasted the savory depth of umami and seen the magnetic strength of the salt bridge. We have moved from the Velcro of Serine and Threonine to the magnets of Aspartic and Glutamic acid. Our molecular landscape is now charged, energetic, and highly communicative. We have explored the acids, the ones who give away a proton to gain a spark. But what happens if we take these powerful magnets and dampen their charge? What if we modify the side chain just enough to keep the social nature of the molecule, but remove the electric buzz? Let's set that idea down now, fully formed, and let our attention drift toward their quieter, more neutral cousins. Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about a pair of amino acids that look almost exactly like the acids we just met, but carry a very different kind of energy. We are moving toward the amides, the molecules that serve as the quiet couriers and diplomats of the nitrogen world. We are headed toward asparagine and glutamine, the ones who help us manage the waste of life with grace and steady reliability. But for now, let the electric sparks of the acids settle into a gentle glow. Feel the magnetic stability of your own structure, and let that sense of biological power carry you deeper into the quiet. Asparagine and glutamine. In our last quiet session, we felt the electric charge of the acids. We watched as aspartate and glutamate crackled with a negative pull, forming the strong magnetic salt bridges that bolt our proteins together and driving the very symphony of our thoughts. But as we continue our journey, we encounter a beautiful piece of molecular editing. What happens if we take those powerful magnets and dampen their buzz? What if we keep the shape and the social nature of the molecule, but remove the sharp electric edge? This brings us to the amides, asparagine and glutamine. Imagine the side chain of aspartate or glutamate once more. At the very tip, where that negatively charged oxygen atom sat, we perform a clever chemical swap. We replace that oxygen with a nitrogen group, specifically an amine group. This creates what chemists call an amide. The transformation is profound. By making this single substitution, the molecule loses its full negative charge. It becomes uncharged, yet it remains highly polar. Think of it as a bell that has been dampened with a soft cloth. The bell still has its shape, and it can still vibrate with the energy of the water around it, but it no longer rings out with a piercing, electric cry. Let us look first at asparagine, the cousin of aspartate. Asparagine holds a special place in the history of our science because it was the very first amino acid ever discovered. In the year 1806, two French chemists, Louis-Nicolas Vauquelin and Pierre-Jean Robiquet, isolated crystals of this substance from the juice of young asparagus shoots. If you have ever enjoyed the earthy, green taste of asparagus, you have encountered the namesake of this quiet traveler. In the world of a protein, asparagine is a master diplomat. Because it is polar but neutral, it can form intricate hydrogen bonds without the aggressive pull of a full charge. It is often the site of a vital biological decoration called N-linked glycosylation. Imagine a protein as a package being sent through a vast postal system. For that package to reach the correct destination, it needs a clear, sticky address label. Your cells create these labels by attaching complex sugar molecules to the nitrogen atom of an asparagine side chain. Without this amide anchor, many of your proteins would never find their way to the surface of the cell or into the bloodstream where they are needed. Asparagine is the quiet scribe, holding on to the information that guides your biology's logistics. Now, let our attention drift toward its larger sibling, glutamine. If asparagine is the scribe, glutamine is the great courier. In your bloodstream, glutamine is the most abundant amino acid of all. It is the body's primary nitrogen shuttle. Nitrogen is an essential element for life. It is the core of our DNA and the heart of every amino acid. But in its free form, as ammonia, it is incredibly toxic. If ammonia were allowed to build up in your blood, it would be like a leak in a chemical factory, causing damage to your brain and your tissues. Your body solves this problem with the grace of a master diplomat. When there is excess nitrogen in your muscles or your brain, your cells package it into glutamic acid, turning it into glutamine. By adding that second nitrogen to form the amide group, the molecule becomes a safe, neutral container for hazardous waste. Glutamine then travels through your veins like a quiet, reliable delivery truck, carrying its nitrogen cargo safely to the liver or the kidneys. Once it arrives, the cargo is unloaded and prepared for disposal, and the glutamine is recycled. It is a tireless, rhythmic service that keeps your internal environment clean and balanced while you sleep. Without this specialized amide transport, the very process of living would become poisonous to us. There is a deeper layer to glutamine that touches our very resilience. During times of great stress or recovery from injury, your body's demand for glutamine skyrockets. It is a primary fuel for the cells of your immune system and the lining of your gut. We often call it a conditionally essential amino acid, because while we can usually make enough of it, there are moments in life where we need more than our internal factories can provide. It is a molecule of support, stepping in to provide the energy and the raw materials needed to rebuild after a storm. It reminds us that even the most neutral members of a community are often the ones doing the most vital work behind the scenes. We can see a fascinating real-world connection to these amides in the way we experience taste and metabolism. While we spoke of glutamate as the source of umami, its amide cousin, glutamine, is much more subtle. It doesn't trigger the same intense, savory signal, but it is deeply involved in how your body manages its cravings and its energy. In some circles of nutrition, glutamine is used to help curb the desire for sugar, likely because of its role as a bridge molecule that the brain can use for energy when glucose is low. It is a stabilizer, a quiet hand on the tiller of your metabolism, ensuring that your energy levels don't swing too wildly from peak to valley. As you lie here, your body is a masterpiece of amide diplomacy. Your liver and kidneys are working in harmony with the glutamine in your blood, quietly processing the day's nitrogen waste. Your cells are checking the sugar address labels on your newly built proteins, ensuring every molecule is headed exactly where it belongs. The electric buzz of the day is fading, replaced by the steady, neutral reliability of these polar workers. They are ensuring that your internal world remains a safe, clean, and well-organized place for you to inhabit. There is a profound sense of order in the existence of asparagine and glutamine. They teach us that sometimes the best way to handle power is to dampen it. They show us that by removing a charge, we can become more versatile couriers and more effective diplomats. They remind us that for a system to be sustainable, it must have a way to transport its most precious and dangerous resources without causing harm. We have felt the dampening of the electric bell and watched the address labels being written by asparagine. We have followed the nitrogen courier, glutamine, through the liquid highways of the blood and seen the history of the asparagus juice discovery. We have moved from the intense, magnetic pull of the acids to the quiet, steady reliability of the amides. Our molecular landscape is now organized, balanced, and safe. We have explored the molecules that manage the waste and the logistics of life. But as we have seen time and again, our journey is one of shifting charges. We have explored the neutral, the hydrophobic, and the negative. Now, we must turn our gaze toward the opposite side of the electric spectrum. We have seen the molecules that give away a proton to become negative. What about the ones that reach out to grab a proton and hold on to it with a positive force? Before we rest here, let me whisper where we're headed next. Not to rush, but to plant a seed about a trio of molecules that carry the basic or alkaline energy of the world. We are moving toward the positive chains, beginning with lysine. We are headed toward the world of long-reaching arms and magnetic attractions that organize the very library of your DNA. We are leaving the neutral diplomacy of the amides and entering a realm of strong, positive connections. But for now, let the quiet work of asparagine and glutamine continue. Feel the balance they bring to your blood and your proteins, and let that sense of steady, rhythmic maintenance carry you deeper into the quiet of the night. Lysine. In our last quiet session together, we ended with the idea of the neutral diplomacy of the amides, the way asparagine and glutamine act as uncharged couriers, managing the nitrogen waste and protein labels of the body with a steady, quiet hand. We saw how a simple swap of an atom could dampen the electric buzz of the acids. But as we move forward tonight, we turn our gaze toward the opposite side of the electric spectrum. We have seen the molecules that give away a proton to become negative. Now, we meet the ones that reach out to grab a proton and hold on to it with a firm, positive force. We are entering the realm of the basic amino acids, beginning with the long-reaching arm of lysine. Imagine, if you will, the central alpha carbon that has been our steady companion throughout this journey. In the side chain of lysine, we see a structure unlike any other we have encountered so far. It is not a tiny block or a massive ring, but a long, flexible chain of 4 carbon atoms stretching out like a molecular arm. At the very tip of this arm is a second amino group, a nitrogen atom bonded to 3 hydrogen atoms. Because this nitrogen is at the very end of the chain, far away from the alpha carbon, it has a unique freedom of movement. It can reach across great molecular distances to find what it is looking for. And in the watery, slightly acidic environment of your cells, this arm carries a permanent, vibrant, positive charge. Think of lysine as a long, magnetic hook, while the acids we studied earlier, like glutamate, were negative sparks. Lysine is their attractive counterpart. It is the seeker of the negative. This positive charge isn't just a chemical detail. It is the fundamental reason you can exist in your current form. To understand why, we must look at the way your body organizes its most precious information. your DNA. If you were to take the DNA from just one of your cells and stretch it out, it would be nearly 2 meters long. Yet, this vast library must fit into a nucleus so small it cannot be seen without a powerful microscope. To solve this problem, your body uses a masterpiece of organization. It wraps the DNA around specialized proteins called histones. Now, DNA carries a very strong negative charge along its entire length. To hold on to that negative thread and wind it up without it becoming a tangled, repulsive mess, the histones must be incredibly positive. This is where lysine shines. Histones are packed with lysine molecules, their long, positive arms reaching out to grab the negative backbone of the DNA. They act like the magnetic velvet on a spool, allowing the thread of your genetic code to wrap tightly and neatly into place. Without the positive reach of lysine, the library of your life would be a chaotic, inaccessible jumble. But lysine's work as a structural organizer goes beyond the nucleus. It is also a master of cross-linking. We have spoken often of collagen, the protein that gives your skin its elasticity and your bones their strength. While cysteine uses sulfur handshakes and proline uses rigid turns, lysine provides the heavy-duty bolts. Through a process facilitated by an enzyme called lysyloxidase, the side chains of lysine molecules on neighboring collagen strands are modified so they can bond directly to one another. Imagine 2 long ropes lying side by side. By themselves, they are strong, but if you drive steel bolts through them to join them together, they become an unyielding cable. These lysine crosslinks are what give your tendons the power to pull against your muscles and your skin the ability to snap back into place. Interestingly, this process requires the presence of vitamin C, much like the Proline modification we discussed. It is a reminder that the strength of our physical frame is a collaborative effort between the building blocks we are born with and the nutrients we invite in from the world. There is another, more energetic side to lysine that touches the very way you burn fuel. Lysine is the primary precursor for a molecule called carnitine. If your cells are a vast city, the mitochondria are the power plants where energy is produced. But the fuel for these power plants, long-chain fatty acids, cannot simply walk through the door of the mitochondria. They are too large and complex. They need a specialized escort. Carnitine, built from the frame of lysine, acts as this escort. It grabs the fatty acids and carries them across the mitochondrial membrane, ensuring that your body can tap into its fat stores for energy. Whether you are running a marathon or simply maintaining your body temperature while you sleep, you are relying on the shuttle service provided by the children of lysine. Because lysine is an essential amino acid, we must find it in our environment. It is famously lacking in many cereal grains like wheat and corn, which is why cultures around the world have intuitively paired grains with legumes, beans, lentils, and peas, which are rich in lysine. This ancient culinary wisdom ensures that the body has the positive arms it needs to wrap its DNA and bolt its collagen together. It is a beautiful thought to realize that a simple bowl of rice and beans is actually a sophisticated kit for molecular engineering. As you lie here, your body is deep in this work. Your histones are maintaining the perfect tension on your DNA, keeping your genetic instructions safe and organized. Your collagen is being reinforced, its lysine bolts tightening to ensure you wake up feeling whole and strong. And your carnitine shuttles are working quietly in the background, moving fuel into your cellular furnaces to keep the slow, steady fire of your metabolism burning through the night. There is a profound sense of attraction in the story of lysine. It teaches us that for things to be organized, there must be a reach. It shows us that strength doesn't just come from individual pieces, but from the way those pieces are bolted together. It reminds us that our very ability to store information and produce energy depends on a long, flexible arm with a positive heart. We have felt the reach of the four-carbon chain and watched the magnetic spooling of the DNA. We have seen the structural bolts in the collagen and followed the carnitine shuttle into the heart of the mitochondria. We have moved from the neutral diplomacy of the amides to the positive attraction of lysine. Our molecular landscape is now reinforced with long-range connections and organizational strength. We have explored the first of the basic travelers, the one who reaches out to bring order to the chaos. And with that understanding of positive attraction quietly in place, we're naturally led to ask, is there a molecule even more basic, even more positive than lysine? If lysine is a single magnetic hook, what happens when we look at a traveler who carries a triple thread of nitrogen, a molecule so alkaline and so attractive that it acts as the heavyweight champion of the basic world? Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed about arginine. We are moving toward a giant, a molecule that doesn't just hold a charge, but serves as a guardian of your blood flow and a master of the waste cycle. We are leaving the long arm of lysine and entering the realm of the most alkaline side chain in all of biology. But for now, let the positive reach of lysine give you a sense of order. Feel the stability of your DNA and the strength of your frame, and let that sense of organized peace carry you deeper into the quiet of the night. Arginine. In our last quiet session, we saw lysine's long-reaching arm, that four-carbon chain with a positive heart that acts as a magnetic spool for your DNA. We felt the power of attraction and the beauty of organization. But now, we meet the heavyweight champion of alkalinity, a traveler so robust and so charged that it stands as the most basic of all the 20 amino acids. This is arginine. If lysine was a long-reaching hook, arginine is a powerful, three-pronged magnetic anchor. To visualize its structure, imagine our central alpha carbon holding onto a three-carbon bridge. At the end of this bridge, however, we don't find a simple amino group. Instead, we find a complex structure called a guanidino group. This group is a masterpiece of nitrogen chemistry, consisting of one carbon atom bonded to three different nitrogen atoms. Because of the way these nitrogens share their electrons, a process we call resonance, the positive charge is not stuck in one place. Instead, it hums and vibrates across the entire group, making it incredibly stable and almost impossible to neutralize. Think of arginine as a lighthouse of alkalinity in the slightly acidic sea of the cell. In the standard environment of your body, arginine never lets go of its proton. It remains stubbornly, faithfully positive. Because of this intense basic nature, arginine is often found at the very heart of the most critical biological interactions. It is the molecule that forms the strongest salt bridges, snapping together with the negative acids like aspartate to lock a protein into a shape that can withstand heat, pressure, and time. Arginine is the giant that holds the ceiling of the molecular world aloft. To understand the deeper layer of arginine's purpose, we must look at its role as a protector. Inside your body, the process of breaking down proteins for energy produces a byproduct called ammonia. Ammonia is a harsh, toxic substance, a chemical ghost that can wander into your brain and cause confusion or even deep sleep from which it is hard to wake. Your body cannot allow this. It needs a way to capture the ammonia and turn it into something harmless. This is the urea cycle, and arginine is the star of this vital cleanup crew. Imagine a grand, circular assembly line. At one point in the circle, a molecule called ornithine accepts a piece of waste. As the line moves, that molecule is transformed, step by steady step, until it becomes arginine. At this precise moment, an enzyme called arginase steps in and clips a piece off the arginine molecule. That discarded piece is urea, a completely safe, water-soluble compound that your kidneys can easily wash away. The remaining part of the molecule becomes ornithine again, ready to start the cycle over. Arginine is the final, triumphant stage of this waste management miracle. It is the guardian that ensures the exhaust of your metabolism doesn't become a poison. Every time you breathe, every time you move, arginine is working in the background to keep your internal sky clear of the clouds of ammonia. But there is a second, perhaps even more poetic side to arginine. It is the precursor to a tiny, miraculous gas called nitric oxide. For a long time, scientists thought gases were just waste products. But in the late 20th century, they discovered that nitric oxide is actually a profound messenger. When your body needs to increase blood flow, perhaps to warm your hands, or to send more oxygen to your heart, an enzyme takes a single nitrogen atom from the side chain of arginine and combines it with oxygen. This tiny puff of gas drifts into the walls of your blood vessels and tells the smooth muscle cells there to relax. Imagine a tight, narrow garden hose suddenly becoming wide and soft. This is vasodilation. It lowers your blood pressure and allows the river of your life to flow more easily. This discovery was so significant that it earned a Nobel Prize, and it all begins with the side chain of arginine. When you feel a sense of warmth and relaxation spreading through your limbs as you drift towards sleep, you are experiencing the chemical legacy of arginine. It is the molecule that tells your body it is safe to open the pathways and let the current move without resistance. There is also a fascinating cross-topic connection between arginine and the way we grow. Arginine is known as a secretagogue, a word that sounds like a mystery but simply means it encourages the release of other substances. Specifically, arginine can stimulate the pituitary gland to release growth hormone. This is why it is often studied in the context of childhood development and tissue repair. It seems that this basic giant isn't just about holding things together or cleaning up waste. It is also about building the future. It provides the signal that tells your body it has enough resources to grow taller, stronger, and more resilient. As you lie here, arginine is performing its dual role as the protector and the relaxer. In your liver, it is completing the final turn of the urea cycle, ensuring your blood is clean and pure. In the lining of your arteries, it is giving up its nitrogen to create the nitric oxide that is currently easing the tension in your chest and slowing your heart rate to a peaceful, resting rhythm. You are a vessel of these quiet, alkaline transitions. There is a profound dignity in arginine. It teaches us that strength and gentleness are not mutually exclusive. It is the most basic and powerfully charged of all the amino acids, yet its primary jobs are to clean up toxic waste and to tell our blood vessels to relax. It shows us that true power often manifests as a service to the whole, as a way to maintain the flow and protect the environment. It is the structural anchor that also knows how to let go. We have felt the vibrations of the guanidino group and watched the assembly line of the urea cycle. We have followed the puff of nitric oxide gas as it opens the highways of the blood and seen the signal of the growth hormone. We have moved from the long, reaching arm of lysine to the three-pronged anchor of arginine. Our molecular landscape is now clean, relaxed, and structurally sound. We have explored the giants of the basic world, the ones who hold on to their protons and their purpose with unyielding strength. But as we have seen time and again in our journey, nature loves to find a middle ground. We have seen the molecules that are always positive and those that are always negative. But what happens when an amino acid doesn't just hold a charge, but can change its mind depending on its environment? What if a molecule could flick between being charged and being neutral with just a tiny shift in the world around it? So what happens when an amino acid doesn't just hold a charge, but can change its mind depending on its environment? That's the question we'll gently explore next as we meet histidine, the shapeshifter. We are moving toward a molecule that acts as a pH-sensitive switch, a traveler that can grab a proton and let it go with the grace of a relay runner. We are leaving the unyielding alkalinity of arginine and entering the realm of the most versatile catalyst in your biological toolkit. But for now, let the basic giant arginine keep you safe and relaxed. Feel the clean flow of your blood and the stability of your structure, and let that sense of protected peace carry you deeper into the quiet of the night. Histidine. We just saw the unyielding alkalinity of arginine, that basic giant that stands as a powerful, permanent anchor of positive charge. We felt the strength of its three-pronged magnetic pull and the peace it brings to our blood flow. But as we move forward, we encounter a traveler that is far more mysterious and adaptable. While arginine and lysine are steadfast in their positive nature, this next amino acid is a master of disguise, a molecular shapeshifter that can change its character based entirely on the world around it. This is histidine, the pH switch of the biological world. To understand histidine, we must look at its unique side chain, the imidazole ring. Imagine a five-membered ring containing two nitrogen atoms nestled like a small, intricate key within the larger structure of the molecule. What makes this ring so special is a property chemists call its pKa. Every amino acid has a pKa, a specific tipping point on the scale of acidity and alkalinity. For most amino acids, this tipping point is far away from the natural pH of your body, which sits at a slightly alkaline 7.4. Arginine's tipping point is way up at 12, so it stays positive. Aspartate's is down at 4, so it stays negative. But histidine's tipping point is approximately 6. This number is very close to the neutral 7 of pure water and the 7.4 of your blood. Because histidine lives so close to its own tipping point, it exists in a state of constant, delicate balance. Imagine a person standing perfectly poised on a balance beam. A tiny nudge to the left, and they lean one way. A tiny nudge to the right, and they lean the other. Histidine is that person. In an environment that is just a little more acidic, histidine grabs a proton and becomes positively charged. In an environment that is just a little more alkaline, it lets the proton go and becomes neutral. It is the only amino acid that can flick between being a charged magnet and a neutral diplomat within the normal range of human life. This ability to change on the fly makes histidine the ultimate catalyst. In the active sites of your enzymes, those tiny biological machines that build and break molecules, histidine often acts like a relay runner. Because it can grab a proton and release it so easily, it functions as a proton shuttle. It might reach out to grab a hydrogen ion from one part of a reaction, and in the next microsecond, hand it off to another. It is the master of the acid-base dance. Without histidine's agile hands, many of the chemical reactions that keep you alive, including the way your body processes energy from food, would move far too slowly to sustain life. It provides the rhythm and the speed to the metabolic symphony. Beyond the world of enzymes, histidine plays a heroic role in a molecule you rely on with every single heartbeat: hemoglobin. You know hemoglobin as the protein that carries oxygen from your lungs to your toes. But how does it know when to hold on to that oxygen and when to let it go? The secret lies in two specific histidine residues, often called the proximal and distal histidines. Imagine a tiny iron cradle inside the hemoglobin molecule, waiting to catch an oxygen atom. One histidine sits below the cradle, holding the iron in place like a steady hand. The other sits above the cradle, acting like a gatekeeper. When you are in your lungs, where the environment is slightly more alkaline, these histidines help the iron grip the oxygen tightly. But when the hemoglobin reaches your hard-working muscles, which are slightly more acidic because they are producing carbon dioxide, the histidines sense that acidity. They change their charge, causing the hemoglobin to shift its shape and gently drop the oxygen exactly where it is needed. This is the Bohr effect, a masterpiece of molecular sensing. Histidine is the reason your breath can reach your muscles. It is the sensor that ensures the fuel of life is delivered with perfect timing. There is also a fascinating, more earthly side to this molecule that you have likely experienced on a spring day or when you are around a frisky pet. Histidine is the direct precursor to histamine. Through a simple chemical reaction where a carboxyl group is removed, the quiet amino acid histidine is transformed into the powerful signaling molecule histamine. While histidine helps you breathe and think, its cousin histamine is the alarm bell of your immune system. It is the chemical that causes your blood vessels to leak and your tissues to swell when your body thinks it is under attack. When you have an allergy and you take an antihistamine, you are essentially trying to quiet the message that was built from the frame of histidine. It is a reminder that in our biology, the most useful tools are often the most potent. The same imidazole ring that shuttles protons and delivers oxygen can also sound a loud, systemic alarm. It shows us that histidine is truly a molecule of communication, whether it is whispering to a proton in an enzyme or shouting to the immune system during an allergic reaction. Even in our diet, histidine holds a unique position. For adults, we can often make enough histidine to get by. But for growing children, it is considered an essential amino acid. Their rapidly developing bodies and growing stores of hemoglobin require more histidine than their internal factories can produce. It is found in abundance in foods like fish, meat, and whole grains. When we eat these things, we are gathering the shifters and the sensors that will keep our internal chemistry agile and responsive to the world. As you lie here, your histidine molecules are performing their quiet, shape-shifting work. In your red blood cells, they are sensing the subtle shifts in pH, ensuring your brain and your heart are receiving a steady supply of oxygen while you rest. In your enzymes, they are shuttling protons back and forth with the speed of light, maintaining the low-voltage hum of your resting metabolism. They are the versatile actors in your internal theater, ready to change their role in an instant to keep the play moving forward. There is a profound lesson in the existence of histidine. It teaches us that adaptability is a form of strength. While arginine is strong because it never changes, histidine is strong because it can be whatever the environment requires. It shows us that the most effective way to manage a complex system is to have switches that can sense the subtle changes in the wind and respond accordingly. It is the molecule of the middle ground, the one that bridges the gap between the positive and the neutral. We have felt the balance of the imidazole ring and watched the relay race of the proton shuttle. We have followed the sensing gate in the hemoglobin molecule and seen the alarm bell of histamine. We have moved from the unyielding alkalinity of arginine to the agile, pH-sensitive switch of histidine. Our molecular landscape is now responsive, balanced, and incredibly precise. We have explored the charged, the polar, and the aromatic travelers. We have met all 20 of the individuals who make up the alphabet of your life. But as we have seen time and again, these 20 individuals do not exist in isolation. They are part of a larger story, a story of how we acquire them and how they come together. We have finished our look at the individual letters. Now, we must look at the ink and the paper. We must step back and see how these 20 different personalities are balanced within the grand economy of your nutrition. Let's set that idea down now, fully formed, and let our attention drift toward the bigger picture, how we actually acquire these 20 different building blocks. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed about the balance between the ones we can make and the ones we must find. We are moving toward the distinction between the essential and the non-essential. We are headed toward the nutritional harmony that connects your body to the wider world. But for now, let the shape-shifting peace of histidine settle over you. Feel the agile balance of your own internal chemistry, and let that sense of responsive harmony carry you deeper into the quiet of the night. Essential versus non-essential. In our last quiet moment, we spoke of the magic of histidine, that agile shapeshifter that senses the subtle shifts in our internal chemistry and adjusts its charge to keep the rhythm of our breath and our enzymes in perfect time. We have now walked through the entire gallery of the 20 travelers. We have seen the small, the large, the charged, and the neutral. But as we look back at this complete set of 20, a new question emerges, a question not of structure, but of origin. Where do these molecules come from? How does your body, in its infinite wisdom, ensure that all 20 are present at the exact moment a new protein needs to be born? This brings us to the quiet, vital balance of the essential and the non-essential amino acids. In the world of biochemistry, we use the word essential in a very specific way. It does not mean that these amino acids are more important than the others. Your body needs all 20 to function, and if even the lowliest glycine were missing, the great structures of your life would begin to falter. Instead, essential refers to a biological boundary. It marks the limit of what your own internal factories can create. Of the 20 travelers, there are 9 that your body cannot build from scratch. You lack the genetic blueprints and the complex metabolic machinery to forge their specific side chains. These 9, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine, must be invited into your body from the world outside. They must be gathered from the food you eat. Imagine, if you will, a master builder who is commissioned to construct a magnificent, sprawling estate. This builder has a workshop filled with every tool imaginable. He can forge his own nails, he can mix his own mortar, and he can even carve simple wooden planks from the trees in his own backyard. These are like the non-essential amino acids, the ones your body can synthesize by rearranging the carbon and nitrogen it already has on hand. But even the most skilled master builder cannot manufacture everything. For the large, intricate stained glass windows, the heavy steel beams, or the rare, fragrant cedar for the roof, he must look beyond his own workshop. He must order these specialty materials from a master craftsman who lives in a distant forest. These are the essential amino acids. Your body is the builder, and the natural world is that distant forest. There is a profound evolutionary reason for this dependency. Creating a complex molecule like tryptophan with its massive double indole ring, or phenylalanine, with its heavy aromatic core, requires a tremendous amount of energy. It takes many steps, many enzymes, and many molecules of ATP, the currency of cellular energy, to build these structures from the basic elements of the earth. Long ago, our ancestors realized that plants and certain bacteria were already doing this heavy lifting. These organisms use the power of the sun to forge these complex rings and chains. Evolution, in its ruthless efficiency, decided that it was better for us to lose the ability to make these expensive molecules ourselves. Why maintain a costly, high-energy factory in every cell when you can simply import the finished product from the environment? This choice made us more energetic and more mobile, but it also made us fundamentally connected to the web of life. We are, at our most basic molecular level, a collaborative project with the Earth. This balance leads us to the concept of complete proteins. When we speak of a complete protein, we are describing a source of food that contains all 9 of those essential specialty materials in the correct proportions. Most animal-based foods, meat, fish, eggs, and dairy, are complete. They provide the entire kit of 20 amino acids in one package. But the plant world offers a different kind of harmony. Many plants are incomplete on their own. Perhaps a grain of rice is low in lysine, while a bean is low in methionine. But when they are eaten together, they complement each other perfectly. The missing keys of one are provided by the other. This is the biological reality behind the ancient culinary wisdom found in every corner of the globe. Rice and beans in the Americas, soy and rice in Asia, lentils and wheat in the Middle East. Your body doesn't care if the 20 travelers arrive in one boat or two, as long as they are all present when the work of building begins. But there is a third category that whispers of your body's incredible adaptability: the conditionally essential amino acids. These are molecules like arginine and glutamine, which we have already met. Under normal, peaceful circumstances, your body can manufacture enough of these to meet its needs. But during times of great transition, during rapid growth in childhood, after a physical injury, or when the immune system is fighting a long battle, your internal demand for these molecules can outstrip your ability to make them. In these moments, your non-essential workers become essential guests. Your body recognizes its own limits and asks for help from the outside. It is a beautiful example of biological humility. The understanding that there are times when we must rely on more than just ourselves to heal and grow. As you lie here, your body is managing this vast nutritional economy with effortless grace. It is sorting through the travelers you have welcomed in today, sending the essentials to the liver to be distributed, and using the non-essentials to fill in the gaps. It is like a busy port at night, where crates are being unloaded, checked against a manifest, and sent off to the construction sites of your cells. The methionine you found in a seed is being sent to start a new protein chain. The lysine from a bean is being sent to reinforce your collagen. The glutamine your body made is being sent to fuel your immune cells. You are a living testimony to the balance between self-sufficiency and dependency. Consider the non-essential travelers for a moment, the 11 individuals like Alanine, Serine, and Aspartate that you can make for yourself. These are often built through a process called transamination. Your body takes a carbon skeleton from the breakdown of sugars and shuffles a nitrogen group onto it from another amino acid. It is a game of molecular musical chairs, where the pieces are rearranged until the correct shape is formed. This allows your body to be flexible. If you have too much of one and not enough of another, you can simply transform the excess into what you lack. You are your own internal alchemist, turning one form of life into another. This nutritional harmony is the silent backdrop to your existence. It is the reason you can recover from a long day and wake up renewed. It is the reason a simple meal can feel so deeply restorative. You are not just eating. You are gathering the alphabet of your own story. You are ensuring that when the ribosome in your cell begins to read the instructions for a new muscle fiber or a new antibody, it doesn't find a blank space where an essential traveler should be. You are providing the materials for the master builder to continue his work. There is a profound sense of peace in this connection. It teaches us that we do not have to carry the whole world within ourselves. We are allowed to be incomplete because the world around us is full of what we lack. We are designed to be dependent, to be part of a larger cycle of energy and matter. The essential 9 are the cords that tether us to the plants, the soil, and the sun. They remind us that we are guests at a much larger table. We have explored the 9 essential guests and the 11 internal craftsmen. We have seen the mastery of the builder and the necessity of the distant forest. We have followed the logic of evolutionary economy and the harmony of complementary diets. We have moved from the individual personalities of the 20 amino acids to the grand economy of how we gather them. Our molecular landscape is now fully populated, fueled, and balanced. We have the bricks, and we know where they come from. But as we have seen, bricks alone do not make a cathedral. A collection of individual travelers, no matter how balanced, is just a crowd. To become life, they must be joined. They must be linked together in a specific, rhythmic order that never breaks. We have reached the end of our look at the individuals. Now, we must look at the bond that makes them a team. We must move from the what to the how. Before we rest here, let me whisper where we're headed next, from the individual bricks to the mortar that holds them together. We are moving toward the peptide bond, the strong, dehydration-driven connection that turns a list of letters into a living sentence. We are leaving the world of nutritional balance and entering the world of molecular construction. We are headed toward the backbone of life, the rhythmic chain that allows these 20 diverse personalities to become a single, breathing whole. But for now, let the nutritional harmony of your body settle you into a deep sense of belonging. Feel the connection between your cells and the world outside, and let that sense of balanced peace carry you deeper into the quiet of the night. The peptide bond. We have spent our time together looking at the 9 essential guests and the 11 internal craftsmen, those 20 diverse personalities that make up the alphabet of your existence. We have seen where they come from and how your body maintains the grand nutritional harmony that keeps them in balance. But a collection of individual travelers, no matter how perfectly matched, is still just a crowd. A pile of bricks, no matter how sturdy, is not yet a cathedral. To transform these 20 distinct characters into the living, breathing machinery of your life, they must be joined. They must be linked together in a specific, rhythmic, and unbreakable order. This brings us to the very ink of our biological story, the peptide bond. Imagine two of our travelers standing side by side. On the left, we have one amino acid, reaching out with its acidic carboxyl group, that red reactive ember we studied at the beginning. On the right, we have another amino acid, reaching out with its basic amino group, that cool, nitrogen-rich blue mist. These two groups are like two puzzle pieces that were designed by the universe specifically to find one another. When they meet, they don't just bump into each other. They perform a beautiful, rhythmic act of creation called dehydration synthesis. In the quiet world of the cell, as these two groups touch, they exhale. The carboxyl group gives up an oxygen and a hydrogen, and the amino group gives up a single hydrogen. Together, these pieces, 2 hydrogens and 1 oxygen, combine to form a single, perfect molecule of water. $H, 2O. This tiny drop of water is released into the cellular sea, a small sacrifice made to allow a greater union. In the space left behind by this exhalation, a new, powerful, covalent bond snaps into place, joining the carbon of the first amino acid directly to the nitrogen of the second. This is the peptide bond. It is a moment of profound transformation. By letting go of a piece of themselves, the two travelers have become something entirely new. They are no longer individual molecules. They are a dipeptide, the first link in a chain that may eventually stretch for hundreds or even thousands of units. To understand the true nature of this bond, we must look at it through the eyes of a master architect. You might imagine a chain as a series of loose, floppy links, like a piece of jewelry that can twist and turn in any direction. But the peptide bond is different. Because of a beautiful quirk of physics called resonance, the electrons in this bond are shared in a way that makes it behave like a partial double bond. Think of the peptide bond not as a flexible string, but as a rigid, flat, wooden board. The carbon, the oxygen, the nitrogen, and the hydrogen involved in the bond all sit on the same flat plane. They cannot twist or rotate. This rigidity is one of the most important secrets of life. If the peptide bond were floppy, your proteins would be like cooked spaghetti, unable to hold a shape, unable to act as a key, and unable to support your weight. But because the bond is a rigid plate, the protein chain has a built-in structure. It can only bend at the hinges, the alpha carbons that sit between the bonds. This creates a predictable, rhythmic movement, like a series of stiff planks joined by flexible joints. It is the perfect balance of strength and maneuverability. This process of joining happens in a very special place, the ribosome. If your cell is a vast library, the ribosome is the master storyteller who sits at the center, reading the instructions from your DNA and typing out the protein sentence. The ribosome is the most ancient and tireless factory in existence. It moves along the genetic code with a steady, clicking rhythm, grabbing the correct amino acid from the cellular crowd, bringing it to the end of the growing chain, and facilitating that exhalation of water. Every second of every day, trillions of these ribosomes are clicking away, forging these peptide bonds with a precision that no human machine can match. They are the weavers of your physical form, turning a list of 20 letters into the long-form narrative of your life. As the ribosome continues its work, a repeating pattern begins to emerge. Nitrogen, carbon, carbon, nitrogen, carbon, carbon, over and over again. This is the polypeptide backbone. It is the silver thread upon which all our diverse travelers are strung. While the R groups, the travelers' backpacks, provide the variety and the function, the backbone provides the continuity. It is a universal structure, identical in every protein, in every living thing on Earth. Whether it is the protein that makes a blade of grass green or the protein that allows you to remember the sound of a loved one's voice, They are all built upon this same rhythmic NCC-NCC sequence. It is the heartbeat of biochemistry, a steady, pulsing foundation that connects all life in a single, unbroken chain. There is a beautiful, real-world connection to this in the way we sustain ourselves. When you eat protein, your body performs this entire process in reverse. In your stomach and small intestine, specialized enzymes use a process called hydrolysis, which literally means water breaking. They take a molecule of water and use it to inhale, forcing it back into the peptide bond. The bond snaps, the two amino acids are released, and they are once again individual travelers, ready to be absorbed into your blood and carried to a ribosome, where they will be used to build something new. It is a perfect circular economy of joining and releasing, a rhythmic recycling of the very alphabet of life. You are, in a very real sense, a collection of amino acids that have been joined and released countless times over the billions of years of life's history on this planet. As you lie here, this grand construction project is moving into its most active phase. While you rest, your ribosomes are working at their peak, weaving the amino acids you gather today into the new proteins your body needs to repair your muscles, fight off infections, and process your memories. The exhalation of water is happening trillions of times over within you, a silent, microscopic mist that signifies the birth of new structures. The rigid plates of the peptide bonds are clicking into place, forming the sturdy backbones of your future health. You are being rewritten while you sleep, one peptide bond at a time. There is a profound sense of peace in the rhythm of the chain. It teaches us that to build something great, we must be willing to let go of a small piece of ourselves to form a bond with another. It shows us that strength comes from a repeating, reliable foundation, a backbone that never wavers. It reminds us that we are part of a continuous narrative, a long, winding chain of life that stretches back to the beginning of time and forward into the morning. We have felt the exhale of the water and watched the rigid plates of the bond snap into place. We have followed the master storyteller, the ribosome, as it weaves the silver thread of the NCC backbone. We have seen the circular beauty of hydrolysis and the rhythmic construction of the sleeping body. We have moved from the individual bricks to the mortar that holds them together. Our molecular landscape is no longer a collection of travelers. It is a series of long, elegant sentences. We have the ink and the paper, and the story is being written. But as we have seen, a sentence is not just a straight line. For a sentence to have meaning, it must be spoken. For a protein to have function, it must have a shape. A long, straight chain, no matter how perfectly forged, is still just a string. To become a living machine, that string must fold, twist, and collapse into a beautiful, three-dimensional masterpiece. Before we rest here, let me whisper where we're headed next, not to rush, but to plant a seed about the final miracle of the amino acid journey. We are moving toward protein folding, the process where the sequence of letters becomes the architecture of life. We are headed toward the moment where the hydrophobic huddle, the sulfur handshake, and the magnetic salt bridge all work together to pull the chain into its final, functional form. But for now, let the steady rhythm of the peptide bond give you a sense of continuity. Feel the strength of the chain that holds you together, and let that sense of rhythmic construction carry you deeper into the quiet of the night. Protein folding. In our last quiet session, we saw the birth of the peptide chain. We watched as the ribosome, that ancient molecular storyteller, clicked along the genetic code to forge the rigid silver thread of the peptide backbone. Let's pick up that thread right where we left it at the moment the string is finished. But the work of life has only just begun. A long, straight chain of amino acids, no matter how perfectly sequenced, is like a list of ingredients that has not yet been cooked, or a pile of lumber that has not yet been framed. To become a living machine, a catalyst, or a structural support, that limp string must undergo the final miracle of biochemistry. It must fold. Imagine, if you will, a long silk ribbon with thousands of tiny magnets, hooks, and velvet patches sewn into its surface. If you were to hold this ribbon high and let it go, it wouldn't simply fall in a tangled heap. Instead, as it falls through the air, the magnets would find their opposites, the hooks would catch their loops, and the velvet patches would cling to one another. By the time it hit the ground, it would have collapsed into a precise, intricate, three-dimensional sculpture. This is protein folding. It is the process where the one-dimensional code of your DNA is translated into the three-dimensional reality of your body. This process happens with a speed and precision that defies human imagination. There is a famous concept in science called Levinthal's paradox. If a medium-sized protein were to try every possible shape and configuration by random chance, it would take longer than the age of the universe to find the right one. And yet, within your cells, proteins fold into their perfect, functional shapes in a fraction of a second. They don't guess. They are guided by the very personalities of the 20 amino acids we have spent our nights studying. The journey from a string to a shape happens in stages, like a grand architectural project. The first stage is what we call the primary structure. This is simply the sequence, the order of the letters: methionine, glycine, alanine, lysine. But almost as soon as the chain emerges from the ribosome, it begins to form its secondary structure. This is where the backbone we studied in Lesson 18 begins to breathe. Because of the way the nitrogen and oxygen atoms in the peptide bonds are arranged, they begin to form hydrogen bonds with one another. The secondary structure, alpha helix and beta pleated sheet of protein molecule that's showing in blue and white concept. This creates two iconic shapes. The first is the alpha helix, a beautiful, winding spiral staircase. Imagine a telephone cord or a delicate vine climbing a trellis. This shape is held together by the rhythmic handshakes of the backbone, providing a spring-like elasticity. The second shape is the beta-pleated sheet. Imagine a piece of paper that has been folded back and forth to create a series of rigid ridges, like a corrugated metal roof. These sheets provide a flat, stable surface that can be incredibly strong. When you look at the silk of a spider's web, you are seeing the strength of these beta sheets, thousands of them stacked together like a molecular shield. As these spirals and sheets form, the protein enters the most dramatic phase of its creation, the tertiary structure. This is often called the hydrophobic collapse. You will remember our study of the hydrophobic travelers, Leucine, Valine, and Isoleucine, the ones who fear the water. As the protein chain floats in the watery sea of the cell, these amino acids are like children caught in a sudden rainstorm. They don't want to be on the outside, so they all huddle together toward the center of the molecule. This huddle is the primary force that drives the folding. The hydrophobic travelers push their way to the middle, creating a dry, oil-loving core, while the polar travelers like serine and the charged ones like glutamate reach outward to touch the water. While this is happening, the safety pins of cysteine lock the folds into place with disulfide bridges, and the magnets of lysine and aspartate snap together in salt bridges. The result is a unique, three-dimensional masterpiece, a globe with its own hills, valleys, and secret pockets. This final shape is what allows an enzyme to recognize a specific sugar or an antibody to recognize a specific virus. In biology, shape is destiny. If the shape is wrong, the machine simply does not work. Sometimes, the folding process is so complex that the protein needs a bit of help. Your cells possess a family of specialized proteins called chaperones. Think of these as molecular nannies or wise mentors. Some chaperones look like little hollow barrels. An unfolded protein chain will wander into the barrel, where it is shielded from the crowded, chaotic environment of the cell. Inside that quiet, protected space, the protein can find its shape without interference. Once the folding is complete, the lid of the barrel opens, and the functional protein steps out, ready to begin its life's work. But what happens when this miracle fails? This brings us to a deeper, more sobering layer of our chemistry. Occasionally, a protein will misfold. It will take on a shape that is not only useless, but sticky. These misfolded proteins can clump together like tangled weeds in a garden, forming plaques that the cell cannot clear away. This is the root of many challenges, such as Alzheimer's disease or the mysterious prions. It is a reminder of how delicate the balance of life truly is. The beauty of our existence depends on trillions of these tiny strings finding their perfect shape every single second, without fail. There is a profound, real-world connection here to the very cutting edge of human knowledge. For decades, predicting how a protein would fold was considered the holy grail of biology. We knew the sequences, but we couldn't see the shapes. But recently, through the marriage of biology and artificial intelligence, we have begun to solve this puzzle. We are finally learning to speak the language of the amino acids, to predict how their 20 different personalities will interact to create the architecture of life. It is like finally being able to look at a blueprint and see the finished cathedral. As you lie here, your body is a silent, bustling construction site of folding. Millions of new proteins are currently collapsing into their final shapes. The alpha helices are winding, the beta sheets are stacking, and the hydrophobic centers are huddling in the dark, dry warmth of your cellular interiors. Your molecular nannies are guiding the young proteins, and your cysteines are pinning the structures into place. You are being shaped from the inside out, a continuous, rhythmic transformation from a simple code into a complex, functional, and living being. There is a sense of wonder in the collapse. It teaches us that our identity is not just in the letters we are given, but in the way we choose to fold them. It shows us that from a simple, linear path, we can create infinite depth and complexity. It reminds us that for anything to be truly useful, it must find its shape, and to find its shape, it must be willing to bend, to huddle, and to lock hands with its neighbors. We have felt the winding of the spiral and the strength of the pleated sheet. We have watched the hydrophobic huddle create the dry heart of the molecule and followed the protein into the protective barrel of the chaperone. We have moved from the rigid peptide bond to the three-dimensional masterpiece of the finished protein. Our molecular landscape is no longer just a collection of sentences. It is a city of machines, a forest of structures, a complete and breathing world. And so we come to the end of our time together, not an ending, really, but a place to rest. You've let these ideas settle beside you as you slept, and they'll be here whenever you return. Until then, sleep well, and carry nothing but the quiet sense that you understand a little more of the world than you did before. The molecular harmony. In our last quiet moment together, we watched as a single protein chain took its shape, collapsing from a limp string into a beautiful, functional masterpiece. We saw how the hydrophobic travelers huddled in the dark, how the cysteines pinned the folds with their silver safety pins, and how the magnetic salt bridges bolted the structure into place. We watched the one-dimensional code of your DNA become the three-dimensional reality of your existence. But as we step back from that single, folded machine, we begin to see the larger picture. We see that a single protein is just one note in a much larger composition. We have traveled from the simple $C, alpha, $carbon, to the complex dance of folding. And now we arrive at the final harmony. The realization that these 20 individuals are not just a list of names, but a rhythmic cooperation that defines the very essence of life. Imagine, if you will, a grand conservatory where 20 different instruments are laid out on a velvet stage. Each instrument has its own unique voice, its own range, and its own special purpose. Some, like the tiny, flexible glycine, are like the soft, high notes of a flute, providing the delicate transitions and the quiet hinges that allow a melody to turn. Others, like the heavy, aromatic tryptophan, are like the deep, resonant thrum of a cello, providing the weight, the stability, and the velvet curtains of the night. There are the percussive, electric sparks of the acids, aspartate and glutamate, driving the rhythm of our thoughts like a steady drumbeat. And there are the long, reaching arms of Lysine and Arginine, the magnetic strings that hold the entire orchestra in tune. By themselves, these instruments are silent. They are potential energy waiting for a hand to play them. But in the hands of your biology, they are woven into a symphony that never stops. This symphony is playing within you at this very moment. It is playing in the way your heart rhythmically contracts, in the way your lungs exchange the breath of the world for the energy of your cells, and in the way your mind is gently processing the very words I am whispering to you. We are not made of static parts. We are made of a continuous, flowing harmony of 20 diverse personalities. Consider the beautiful paradox of our chemistry. We are built from a language that has only 20 letters. Every enzyme that digests your food, every antibody that protects you from illness, every strand of hair on your head, and every pigment in your eyes is written with this same restricted alphabet. It is a testament to the infinite creativity of the universe that such a small set of tools can create the staggering diversity of life on Earth. From the blue whale in the deep ocean to the tiny moss on a forest stone, we are all variations on the same 20 themes. We share this molecular vocabulary with every living thing that has ever existed. When you breathe, you are participating in a cycle of carbon and nitrogen that has been shared by billions of creatures over billions of years. You are never truly alone. You are part of a molecular conversation that spans the entire history of our planet. As we look back over the landscape we have traveled, we can see the logic of the design. We began with the molecular compass, the alpha carbon, which provides the steady center. We met the minimalists and the builders, the structural rebels and the ignition keys. We felt the heat of the sulfur and the fragrance of the aromatics. We touched the sticky Velcro of the hydroxyl workers and the magnetic pull of the charged powerhouses. We followed the quiet diplomats and the basic giants, and we watched the shapeshifter sense the changing tides of our internal sea. Every one of these travelers was necessary. If we were to remove even one, if the universe had forgotten to create the rigid ring of Proline or the long arm of Lysine, the entire symphony would fall into dissonance. The architecture would collapse, the signals would fail, and the fire of life would go out. Your body is a master of this balance. While you lie here in the stillness, Your internal world is a masterpiece of logistics. It is ensuring that the right amino acids are in the right place at the exact microsecond they are needed. It is recycling the old to build the new, exhaling molecules of water to forge peptide bonds, and guiding young proteins into their protective chaperones. It is doing all of this without your conscious effort, a silent and tireless devotion to your existence. You do not have to tell your cysteines to lock hands or your leucines to huddle together. They simply know their craft. They have been practicing this dance for eons, and they perform it with a grace that is beyond our understanding. There is a profound sense of security in this realization. We often feel that we must hold ourselves together, that we must manage every detail of our lives through sheer will. But at the most fundamental level, you are being held together by a harmony that is far older and far wiser than your thoughts. You are supported by the unyielding density of the aromatic core and the flexible resilience of the peptide backbone. You are protected by the sulfur handshakes and energized by the electric sparks of the acids. You are a vessel of an ancient rhythmic wisdom that functions perfectly even when you are at your most still. Think of the ink of our story one last time, that dehydration synthesis, that exhalation of water that joins two travelers into one. It is a beautiful metaphor for the way life works. To build something larger, something more meaningful, we must be willing to let go of a small piece of ourselves. We must be willing to exhale and to bond. This is true of our molecules, and it is true of our lives. We are individual letters, yes, but our purpose is found in the sentences we form and the stories we tell together. The 20 amino acids teach us that diversity is the key to function, and that connection is the key to strength. As the day's memories begin to soften and fade, let yourself feel the weight of this harmony. You are not a collection of disconnected parts. You are a single, breathing masterpiece of biochemical engineering. Every cell in your body is a small cathedral of folding proteins. Every heartbeat is a pulse of molecular timing, and every breath is a renewal of your nutritional balance. The travelers we have met, from the tiny glycine to the massive tryptophan, are your constant companions. They are the guardians of your rest and the architects of your tomorrow. In our time together, we have looked at the individual notes. We have studied the strings, the brass, and the woodwinds of your biology. We have seen how they are tuned and how they are played. But as the lights dim in the conservatory of your mind, the individual instruments fade into the background. What remains is the music itself, the steady, low-frequency hum of a body in perfect equilibrium. It is a music that doesn't need to be loud to be powerful. It is a music of maintenance, of repair, and of quiet growth. Carry this sense of understanding with you as you drift. You are not a mystery to be solved, but a wonder to be experienced. The language of your life is written in a beautiful, consistent code that ensures you are rebuilt bit by bit through the night. The alpha carbon is your anchor, the peptide bond is your thread, and the folding is your shape. You have seen the entire alphabet now, from lesson 1 to lesson 20. You have the map, you have the compass, and you have the travelers. And so, we come to the end of our time together. Not an ending, really, but a place to rest. You've let these ideas settle beside you as you slept, and they'll be here whenever you return. Until then, sleep well and carry nothing but the quiet sense that you understand a little more of the world than you did before. Your molecules are in their place. Your bridges are pinned. Your symphony is playing. All is well. All is in harmony. Sleep now, and let the quiet chemistry of your being take care of the rest. Until we meet again in the morning light, rest deeply, rest well.